Ah, voilà.
Allô, allô, allô. 1, 2, 1. Thanks to take a seat. We're going to start. Mr. Bedel is here only for a few minutes, so we have to let him the floor as soon as possible. Thanks. Welcome to these hybrid days. I will make a deeper introduction in a few minutes, but as Mr. Bedel has got only a few minutes to dedicate us, I let the floor immediately to him. Mr. Bruno Bedel, General Secretary for Investment, France 2030 Program. Mr. Bedel.
Good morning, everyone. I'm delighted to be here. I have the tough task to wake you up for this hybrid day. Not so easy after this wonderful weather in Paris, right? You're here. More seriously, we live a true revolution in the healthcare industry. And when you go through this time where everything is changing, as a government, you have like 3 conditions to succeed. The first one is a will, determination in really digging a field and effectively deliver and help to deliver solutions. This will is strong in France. Second is a way. A way is a strategy, and I will, in a few minutes, detail a bit of it. But this strategy is necessary to gather all the working forces to make it happen. And third, our souls, or people, understanding and executing this strategy to effectively win and succeed in finding those solutions. And it's based on this that Emmanuel Macron, Mr. Président de la République, has decided 2 years ago to set up le Secrétariat Général pour l'Investissement, General Secretary for Investment, with 54 billion euros in different innovation fields, one of them being health. Health was given 7.5 billion euros to really investigate what could be innovation in health in the future. It's called l'Agence d'Innovation Santé, led by Liz Alter, who will be here, I think, tomorrow, facing you and giving you more details about this agency is organized. So this is showing the will. The way is based on these new strategies, and of course, all the discussion you'll be having during these 2 days are at the heart of it, that effectively we as France want to be part of this bioengineering, biotherapy revolution. and it started already, but it's been given in a very clear form by the president. 2020 biomedicine should be effective by 2030 in France. That's the way that it's drawn. So clearly, instead of just saying, okay, we'll give money to research, or we'll support almost randomly some startups, or we'll give big speeches about it, he preferred to say, this is the goal, and be selective, and SGPI, which I'm running, is extremely selective in the projects we're supporting. It's about 1 of 3 presented that we are selecting for support, different kind of supports, and with the purpose of delivering this 20 biomedicine. Aside from this, we finance for 2 billion euros what we call compétences et métiers d'avenir, so future work skills, which is another strategy where we support any kind of education in this field. It can be new education for students, fresh students. It can be to reform, to give new educations to existing medical experts or chemical experts. So basically, what we try to do at the same time is to find means to support this strategy and to find the people to do it effectively. Because it would be a big mistake to build, walls and laboratories and to build, production facilities. If we're missing the women and the men inside, it won't work. So I don't want to pile up numbers everywhere. I can tell you that already in the course of 2 years, we have engaged at the France 2030. That's the name of the big plan, France 2030. This 54 billion plan. We've already have engaged 30 billion euros. So we're on track in terms of really pushing hard to make things happen, on which probably, on the same percentage probably, we are right now in the health sector, which represent close to 500 million euros supported already by SGPI. We have about 6 biomedicine which are on the way to the different phases, the phase 3 and ready to go. And I'd like to stress one. I'd like to stress one as an example because at a personal level, I was one of these parents who faced the very tough time to have a child who was sick from a genetic disease. And at that time, was 10 years ago, a little more, 15 years ago, there were no solutions, absolutely no solutions. The knowledge at that time was absolutely impossible in terms of complexity to understand this disease, so it was definitely hopeless. And Geniter, which we've been supporting with what we call a biocluster, so it means that we're trying to gather research, academic research, startups, pharmaceutical companies. Geniter recently, I visited Geniter recently after supporting this for a long time. The government did support Geniter for a long time. And it was very emotional for me, but they told me, wow, we found a solution. And it's really important because I want to explain to you that we don't just are governmental people, or we don't just think out of the context of a political competition. Because, on my notes here, the big debate is France ahead of Germany, or is England ahead of Italy? And it doesn't matter, in fact. It's more like the Olympics, and as you know, Paris is welcoming the Olympics. We're all performers. We're all in the same goal to really ease the pain of zillions of people across the world facing new sicknesses, facing new diseases. And we know that through this revolution, through these genetic therapies, we will find ways to cure things that were just impossible to imagine only 10 years ago. And that's really my That's why I wanted to be here this morning, even for 5 minutes, to tell you that if we forget this in the different plans we're making, if the biotech startups forget that the real purpose is not to make money, but the real purpose is to cure people, if we forget that it can only work by the collaboration of people, by the association of ideas, by tests and trials, and failing and bouncing back, if we forget that that's the mission we're in, I think we forget the essential. That you can count on my support, both professionally and personally, to make sure that this 20 biomedicine in 2030 will be real in France for sure. Thank you very much.
Okay, thank you. Thank you very much, Mr. Bunnell, for this very positive introduction. So, welcome to these hybrid days, as I told you, 2 days co-organized by ARIS, the Health Industry Alliance for Research and Innovation, BPI France, the French Public Investment Bank, France Biotech, French Trade Association Biotechnologies, and Inserm, French National Institute of Health and Medical Research, and of course, LEM, which is the French Union of Pharmaceutical Industry. And under, it's written up there, under the high patronage of Emmanuel Macron, our French president of the Republic. So the idea of these 2 days is they all gathered, these organizers, to offer an international research and innovation meeting, bringing together, as you will listen to them, academic researchers, startups, and industry here in Paris in this incredible space, as you've discovered. Today's event alternating keynotes, pitches, and B2B meetings. I think it's very important for you to get these B2B meetings. Some of you may have got some positive answers from people to get their B2B meetings, but maybe not scheduled yet. So if your B2B meetings are not scheduled yet, it's normal. There will be some mismatch and problems, so you just have to take the phone number and to set up a direct meeting with the person you have to meet for the B2B meeting. Normally, all the phone numbers will be delivered. So if you don't get a B2B scheduled yet, you just have to phone the person you have to meet, and it should work. Of course, this 2024 edition is dedicated to the next generation of gene and cell therapies. We talk about vector-derived and gene therapy, novel genome editing approaches, nucleic acid therapeutics, cell therapies, next generation of CAR, novel approaches such as exosomes for delivery and short, medium, and long-term tolerance and toxicity, and so on. A very, very important program during the Tuesdays. A lot of talks and short talks, and sometimes, of course, debates, roundtables with people and of course, it will be the occasion during the roundtables for you to take the floor, to talk, to ask your own questions. So you have some mics, desk mics, you see them just around you. You just have to push and to turn on the mic, push the button when you want to ask a question, and we will hear you very well. And don't forget to turn it off when it's over, when you've asked your question. And it will be there will be several moments to ask your questions, Q&A, questions and answers after a few talks, and also during the roundtables. And I remind you then that the B2B sessions will be organized during the lunch breaks, 2 hours today, 2 hours tomorrow. Today between 1 and 3 p.m. and tomorrow between 12 noon and 2. There's a little change in the schedule towards the program you've got. It's a bit early, as you know, it's a bit earlier. So the session tomorrow between noon and 2, and at the end of the day, at about 5 o'clock, you will have another 2-hour session for your B2B meetings. So there will be 4 sessions, and this morning we'll talk about the challenges associated with future viral vectors and genome editing approaches, with several short talks, overviews, and a roundtable. This afternoon, session 2, new nucleic and therapies applications and vectorization. Session 3, immunocell-based therapies, which will be tomorrow morning. And in the afternoon tomorrow, the 4th and last session dedicated to cell therapies and beyond. To introduce and open the ceremonies now. I let the floor to the various organizers and those who represent them. And the first of them is Thomas Lombès, if you can join me. Maybe the other can come on stage and take a seat too. Thierry Hulot for the LEM, Marc Bonneville for ARIS, please. And maybe if Rosalie Maurice is already here, she can come and sit down too. All right, you've done, yeah. You can take a seat. And the first to speak will be then Thomas Lombès, your Deputy CEO for Strategy at Inserm. Thank you.
Ladies and gentlemen, dear colleagues and friends, I'm Thomas Lombès, Deputy CEO for Strategy at Inserm. And it's my honor to be with you today to address you at this international event on behalf of the French academic research players in the biomedical field. So thank you for joining us for this important occasion, and I'm delighted to see such an audience, including 50 top academic teams, French top academic teams, and over 75 companies ranging from small innovative startups to big pharma companies. So today's topic, gene and cell therapies, is indeed of strategic importance. These therapies represents a major research area and a booming industrial sector with economic impact and also a lot of scientific, technological, and health-related implications. These therapies offer promising avenues for treating a wide range of diseases, but they also come with several challenges. Just to mention a few, efficacy, because achieving consistent and durable therapeutic effects can be challenging. Delivery, because ensuring that a gene or cell reaches the intended site in adequate amounts and remain functional is crucial for the success of the therapy. Safety, because gene and cell therapies can sometimes trigger side effects. So we need to be able to carefully assess potential risks, but also manufacturing and scalability, because answering reproducibility of the manufacturing process while maintaining product consistency and quality is not an easy task. So you would probably have guessed at this point that I'm not an expert on these subjects, but what I know is that we need to collaborate to tackle these challenges. And in France, there is a strong and growing interest among researchers to work with industry and even sometimes create their own companies. And these collaborations are in everyone's interests. For companies, because public research represents a fantastic source of innovation and therefore growth, but for public partners also, because engaging with real-world applications and accessing different expertise is a strong source of inspiration and a driver, often allowing us to push the boundaries of knowledge. Moreover, if we are truly to help patients, scientific and medical advances and discoveries must eventually reach the market, and for this we need industry, of course. So how do we push this a step further? I'm here to tell you France possesses significant assets to compete on the global stage. And first and foremost, the high quality of its academic research, once again, recently highlighted by the European Patent Office, for example. France holds the second position in Europe for clinical trial and early stage research. And I understand that more than 500 therapies are currently in the pipeline. However, I believe that there is still room for growth. I will finish by a word about our President Macron's vision about our national research landscape. As you may know, Inserm is a major biomedical research organization with an annual budget of around 1.2 billion euros and around 350 labs everywhere in France, connected with our partners, university, hospitals, and other academic partners. And President Macron decided to create within our organization a programming agency empowering us to define and implement strategic national biomedical research programs. And you will have, I think, a nice example of what these future programs could be with the 80 million euros research program on biotherapies and biomanufacturing that will be presented tomorrow morning, I think, and that is coordinated by Inserm and Seva. So in conclusion, I urge you to consider France as a strategic partners in research for the next generation of gene and cell therapies. And I hope the discussions over the next 2 days will reinforce this belief, because collaboration and innovation are key to our success. So I wish you an excellent symposium. Thank you very much.
Thank you very much. Thank you, Thomas Lombès. And now I let the floor to Thierry Hulot, chairman of LEM.
Morning to you all. Well, you may wonder what's happened to me wrong this morning. I joined this conference to have partnership between research people, and all of a sudden at 9 a.m. in the morning, you have the head of the Trade Association for Pharmaceuticals. Something is going wrong in this room. Actually not, because let me share with you what we call a raison d'être for the entreprise du médicament. We have one goal, only one goal, is to make sure that whenever a patient needs a treatment, he gets it. Whether this treatment exists at your pharmacy shop at the corner of your home, or it needs to be invented. And here we are today, this morning. And to do so, we have a very clear strategy. And I feel moved by what Bruno Bonnell shared with us and said, guys, the money is not the problem here. The problem is to make sure we find the treatment. And he shared a very personal situation regarding one of his own child. And this is where we need to focus our energy, to make sure that we together create the treatment that are still needed. And there's an immense task in front of us. And this is why the number one priority des entreprises du médicament is to make sure that patients get access to treatment. This is the first thing we have to achieve. And of course, we'll do so only if we have a proper economical environment. But this comes only second. And third is important, we are all on this other mission. It's nice to save treatment. We have to rescue a small planet because there's no plan B. So we have to be ethically, socially responsible. So I'm sitting here in front of you today also for a very personal reason. I started my career in the former millennium, where partnership between public and private institutions. From my company, I would have been fired to talk to Inserm early in the morning. They will have raised question, well, you meet a French official at the coffee corner? Are you out of your mind? This was prohibited. Thanks God we have changing. We change all of this. We put the wall down. And this day we have a unique opportunity to exchange ideas, to share ideas, and really to create value. Innovation starts with people talking to each other. And if I have only one pledge or request to you this morning, is make sure that today you will meet someone you never talked with before. You had no intent to share ideas before, because this will create value. And this is why we are so proud at the LEM to be part of organizing these 2 days, to foster exchanges between people. And let's have a dream that out of one of our exchanges today, in 20 years we will say, this is where we started our collaboration and it has created a new treatment, just as Bruno shared. And count on us, les entreprises du médicament, that when you will have found the baby, this molecule that can move things, we will help you and support you to accelerate clinical research, to make sure there's bioproduction facilities around the corner, and to make it up to the finish line to the patient. So it's time for action, it's time for exchanges, and I'm sure the 2 days in front of us will be fantastic. Thanks for your attention.
Thank you. Thank you very much, Mr. Hulot. And now Marc Bonneville, chairman of ARIS.
Okay, so first of all, Let me welcome you on behalf of all the organizers of this symposium. So you have seen all these acronyms. I will try to explain very briefly who we are, in particular, what is ARIS and what is France Biotech. So France Biotech is a professional association created by and for entrepreneurs in the biotech, medtech, and digital health sectors. As a driving force in the health tech innovation ecosystem and a privileged interlocutor with public authorities, France Biotech helps to meet the challenges faced by the health tech sector and to provide tangible solutions to improve the competitiveness and attractiveness of innovative healthcare companies. ARIS, so the Alliance for Research and Innovation of Healthcare Industries, is an association created in 2010, so it was in a mirror of a similar alliance gathering all the public research institutions in France. And so ARIS brings together several major industrial groups in the pharma, the diagnostic, the medtech area. And its mission is to federate industrial and academic players to enhance the attractiveness of research and innovation in France, and most importantly, to foster public and private partnership. And as you know, and as just mentioned by Thierry, establishing a partnership needs to understand each other. And clearly, researchers, academic researchers, and industrial researchers and innovators are clearly complementary. They are not redundant, they are complementary. And it's key to establish this dialogue between the public and private player throughout the innovation process. And this leads actually ARIS in this objective to foster this acculturation, this mutual acculturation, to organize events bringing together all types of players around particular scientific and technological issues. And this is the very purpose of the Hybrid Days event. So these Hybrid Days events have been organized for many years. They are the merger of two former events that were organized by some of the institutions that are mentioned here. And clearly now, this event gathers all the private and public players of the health tech sector in France. So this is a unique opportunity to foster these exchanges between these players, to foster this mutual acculturation, and in particular, to this end, the Hybrid Days are built around presentations and roundtable discussions in order to cross-reference the themes of excellence of academic research with the expectation and needs of industry. In addition, and most importantly also, this event is an opportunity to mobilize global delegation from the world's leading pharma industries, and so to strengthen the international visibility and attractiveness of the French researchers and innovators in a particular biomedical area. Regarding cell and gene therapy, so the topic chosen for the Hybrid 2022-24 event, so this is a vast subject that covers cell and gene-based approaches with diverse indications, notably rare diseases and cancers. And as mentioned by Thomas, there have been recent achievements that I won't have time to flesh out here, but also many, many challenges. And these challenges deal with both upstream and downstream issues that are absolutely key to reducing costs and scaling up these sophisticated treatments. So the design and development of cell and gene therapies require multidisciplinary skills from both academic and industry researchers, and rely on close and constant dialogue between these public and private players throughout the product development process. The French R&D ecosystem in the field is particularly rich and of high quality, and as illustrated by the program of these 3 days, so you will hear some 50 international renowned scientists, as well as many French biotechs, most of which are direct spin-offs from the activities of several academic research centers. So I hope you'll enjoy listening to the speakers and roundtable participants as much as we enjoy getting together this wonderful program. And on behalf of France Biotech, Aris, LEM, Inserm, and BPI France, I wish you fruitful exchanges and many new successful partnerships. Thank you.
Thank you, Marc Bonneville. And to finish these presentations, introductions by the partners, Rosalie Maurice, your Head of Health Innovation Department, BPI France.
Hello, everybody. I'm very happy to be here with you and be part of this great event. I'm Head of Health Innovation Department at the Innovation Direction at BPI France. BPI France is a key player in fostering innovation in France and is a primary operator for France 2030 that Bruno Bonnel was talking about a few minutes ago. A 50 billion plan supported by the government. BPI France plays a crucial role in financing companies, all kinds of companies, especially startups, supporting innovation and fostering economic development. Through the entire life cycle of the startup, from the startup creation, even before the startup creation in the lab with a deep tech plan, to clinical phase and company development. Few numbers. In 2023, BPI France has deployed 600 million euros in grants to finance innovative projects in health sector, biotech, medtech, and e-health. It could be SMEs project, but also private-public projects that work together. We need to have this collaboration. And since 2021, since the beginning of the 2030 plan, 250 million euros have been deployed to support biotherapy and bioproduction companies, and 130 million has been dedicated especially to cell and gene therapy, which is about 30 biotherapy projects. For this event, we have selected a few promising startups to pitch and be present here today. Thanks to Julie Lelouch to our work on this selection. Moreover, we offer various investment funds such as InnoBio, along with loan, guarantees, and support programs to small and medium-sized enterprises. Through this event, we aim to foster fruitful discussion, encourage innovative collaborations, and building lasting relationships. Thank you for being part of this event, and thank you for les organisateurs.
Thank you, Rosalie. Thank you very much. And thanks to everyone. Thank you. You can take another seat if you want to watch the session. Thank you very much for these introductions. So it's time to launch the conferences and short talks and the overview conference to start. The session one dedicated to the challenges associated with future viral vectors and genome editing approaches. After the overview conference by Oumeya Adjali, we'll have 3 short talks, and then you will have time for questions and answers. So you can keep in mind questions you'd like to ask those who are going to present, the 4 presentations to come, and you'll get about 15 to 20 minutes to ask your questions. So if you can keep them, maybe remember them, write them, and then you will have the floor. You will push on the button for the mic, and then you will ask the questions. It will be about 10, 15. Then there will be a coffee break. There will be some other short talks, other questions and answers, and a final round table before the lunch break. But to introduce this session 1, welcome Oumeya Adjali. Oumeya, okay, you can come, thank you. You're from Inserm, and you're going to present gene therapy using AAV viral vectors from vector engineering to preclinical evaluation. Thank you.
So, good morning, everyone. I'm very delighted to be here. I want to thank the organizers for giving me the opportunity as an Inserm research director, so academia, to talk about gene therapy and AAV vectors and where the field is standing on and what are the challenges we're facing to cure patients. So, as you may know, AV viral vectors are widely used today for gene transfer. They are almost the most used vectors in gene therapy, especially for in vivo gene transfer, where they represent about 85% of the used viral vectors. And this is a study published by Map Design about one year and a half. So what are these AV vectors? So they are derived from a parvovirus family, and they have a number of advantages to transform them in viral vectors for gene transfer. They are derived from viruses that are non-pathogenic, or not associated to pathologies, and they are a dependovirus, which means that they are defective in replication alone and that they need dependovirus. So this is like a safety feature very important for gene therapy. One major advantage is that one unique single in vivo administration can allow very long gene transfer, and we have really years and years of follow-up, even in patients today, without major integration. So they remain mainly in episomal forms, despite the fact that now we know that we have some integrative events also, but no associated to oncogenesis so far. One limit now is that they are very small, these vectors, and the encapsulation size is very limited to only 4.7 kilobases, which means that they are not today, as they are, they cannot be used for large genes. But I will talk on that later on. So what is great with AV vector is that we have plenty of natural serotypes and hundreds of variants now in the labs and in the industry, which means that we can choose à la carte, let's say, one serotype or one variant, playing also with the mode of administration to target virtually all the organs. So for instance, if you want to target the brain, we can use CNRS, intrathecal, intracisternal route administration, and AV10 or AV9. If we want to target the muscle, we can use the intrasystemic route of delivery using AV8 and AV9, which are musculotropic and cardiac tropic. And actually, the AV vectors are already in the market, as you may know. To date, we have, 7 products have already been approved. One was withdrawn, the Glibera, the first one, approved. So these are the products approved by either the FDA and or the EMA for ocular, muscular, metabolic disease, as well as hemophilia. The 2 last ones approved in Europe are for hemophilia A and hemophilia B. But those vectors work using very high doses. And this is very true for systemic or intravenous administration. So for instance, for those products, the 2 last ones for hemophilia and the approved product for Duchenne myopathy, LVDs, we need between 10 to the 13 to 10 to the 14 viral genome or virus, recombinant virus per kg, per kilo, which is very huge. And that means that there is still a gap between, let's say, preclinical developments, manufacturing, and the use of these vectors in patients, because there is clearly a lack of specificity and efficacy of these vectors, and we need really to work on that to reduce the doses, to reduce the needs of manufacturing and the cost, of course, because the cost is very high, is more than 1 to 3 million per product approved today in the market, to reduce as well the toxicity and immunogenicity issues. And there are 2 other major limitations. The limit of the encapsulation size, I was talking earlier, which means that we also figure out how we can treat disease where large gene larger than this size are affected or are the targets. And the other one is that the animals, the model animals we have used so far, mice, non-human primates, have not always predicted what happened in patients. So we really need to also continue developing new models of disease and new models for vector screening and evaluation. So, yeah, so I think I can skip this one because I have. In my lab, target for translational research in gene therapy in Nantes, we are working on some of these limitations and developments. I can say that the lab is really in the heart of the objectives of the National Initiative Stratégie d'Accélération des Biothérapies et Bioproduction. The fact is that we're gathering in the same lab, in the same site, scientists from different fields. So we really believe that multidisciplinarity is needed in the field of gene therapy to develop next gene vectors. And so for that, we have not only biologists, but we have also process engineers, bioinformaticians, chemists, biophysicians, immunologists, and this allows really us to cover the whole chain of AAV vector development, from vector design in silico to vector engineering, vector screening and evaluation in preclinical models, process developments to produce those vectors in large scales and move to the patients. And to make the translation possible to patients, we have also been pioneer to certify the whole activities of the lab, including research activities, under the ISO 9001 system to make the data robust for regulatory agencies and pre-INDs and INDs. And so regarding the manufacturing part, we have a facility in the lab called Centre de Production des Vecteurs, which is one among the 8 integrators of the stratégie d'accélération des biothérapies. So we believe that it's very important to, of course, deal with the process itself. So systems of purifications, but we also try to back to really fundamental biology to the cell that is producing AAV vectors, because here again there are some innovations to make on these cells to increase the yield of production and increase the quality of AAV vectors. But now, during the remaining few minutes, I want to talk about the other hurdles, much more vector engineering and preclinical. So for vector engineering, the majority of people have worked, and we are also working in some projects, on molecular vector engineering, which means engineering of the capsid or engineering of the viral genome itself. But one other possibility is to combine different fields, to combine chemistry and biology, and try to generate chemically modified vectors where we use chemistry methods to conjugate ligands, selected, of course, to target a cell or an organ of interest. So we have been working with those methods. We have a chemist, Mathieu Mével, in the lab, working with a chemistry lab in Nantes, academic lab. And we have already provided some proof of concept to target better targeted than native AAV, the ocular, the retina, the liver, the muscle. Now we are working on the joints to treat maybe one day arthritis, and also the CNS, central nervous system. So another thing is that we not only need vector engineering, but we also need methods to characterize those vectors and correlate the efficacy of these new vectors to their safety, the absence of toxicity, but also efficacy. And to do so, we really need to move from the conventional methods, analytical methods used so far, so the qPCR targeting only one very short sequence in the viral genome, to high-throughput sequencing methods that enable a full characterization of the integrity of the viral genome, because this is really also a limit with the manufacturing methods we have today, and also to characterize or look at the DNA contaminants that we will have certainly from the cells that are produced in the AV vectors. So we have been, again, one among the pioneer labs to develop a DNA-seq method to characterize different batches produced from different methods. And we can even compare and have a signature from the different methods and correlate the quality of the batches to safety and efficacy. Now, regarding the preclinical evaluation, there's also still a gap, because the mouse models, well, they are very good to evaluate the efficacy of a vector, but they don't at all predict immunogenicity. This is what we have seen upon translation to large animal models and patients. And as you know, also non-human primates are now very difficult to use and not very ethically possible to use. And so we believe that moving to other animals could be one possibility, one avenue. So, for instance, we have been working in Nantes at the development of transgenic rats model to modelize Duchenne myopathy and also Stargardt disease, which is a macular-ocular genetic disease. And these models, above the mouse model, they have also an advantage is to make possible the evaluation of the surgical methods of route of administration more closely to a large animal model than the mouse models. Another possibility, for instance, is to move from large non-human primate model to the pig and the mini pig. And the mini pig is very promising, looks as very promising for the evaluation of ocular gene therapy products, because it has a retina that is not so far, let's say, from the human retina. The last possibility is to generate humanized mouse models, where we can humanize either one organ or multiple organs. And it's very interesting to evaluate the immunogenicity of AV vectors upon adoptive transfer of human, for instance, cells with pre-existing immunity against AV. Now, we know that animal models are not perfect, and they need additional complementary models. And we are working also, and many others, in the field of gene therapy and AV gene therapy on human organoids using iPS cells from healthy and patients. So these are very interesting because they will have the exactly same genetic background. Maybe not the epigenetic, this is another story, but at least we can have the mutation we want to treat. And of course, those models are not yet fully mature, and there is still a lot of work on this, but they look really promising to screen AV variants. So last direction is how to overcome this limited capacity of encapsulation. So there are different methods. Some methods or some technology are used dual vectors, which means that we can use two vectors with half and half gene, or we can also insert some specific sequences and express half and half protein and make the recombination possible in vivo. But another possibility is to combine next gene therapy strategies. For instance, an engineered vector with increased efficacy to gene editing CRISPR-Cas9 strategies, and I know that there will be some gene editing also discussions. So using CRISPR-Cas9 not to correct a point mutation, but using it with an AV vector to bring and replace a part of the gene where a hotspot of mutation is observed. And we are working within the frame of peer-to-peer funding with great partners on these strategies and with the proof of concept for against Stargardt disease, the macular genetic disease. So to conclude, so AV vectors are really promising. They are on the market. They are treating dramatically diseases such as SMA, for instance. but there are many, many developments yet to do to improve the efficacy, to improve the safety, to reduce the doses and reduce the manufacturing needs. And of course, we need also in parallel to work on much more relevant models to evaluate and screen the new variants, which means that we must go from bench to bedside and back again every time to develop a new next generation of gene therapy vectors. And it is clear that the partnerships, the partnerships between public and private can only foster and accelerate those developments. So I am looking forward to the discussions I will have during these 2 days. I've just, if I can have 10 seconds, just to, as a, let's say a follow-up of these hybrid days, grid days, I just want to ask you to save the date, because as a member of the board of the French Society of Cell and Gene Therapy, I'm the vice president of the society, and also as a member of the scientific committee of map design. We are working together, map design and the SFTCG, to organize a day dedicated to innovations for cell and gene therapies, gathering again industry and academia. So I believe that it could be a very nice opportunity to continue the discussions. So thank you for your attention, and on the behalf of the team, thank you.
Thank you very much, Oumeya Adjali. Thanks a lot for this brilliant introduction. And you see that AAV vectors will be really the focus of several of the short talks this morning, and of course during the roundtable at the end of the morning. So it's very important to get this overview. Thanks a lot. And by the way, right now we're going to talk about AAV vectors again with Buj-Bello. You can join me. Thank you. You will talk about next generation AAV vectors for gene therapy and muscle disorders.
Okay, so good morning, and first I would like to thank the organizers for giving me the opportunity to present some of the work that we are doing in our laboratory, and I would like as well to thank Dr. Adjali for the very nice and comprehensive introduction on the AAVs and the technology itself. So I am going to show you just a few examples of, very concrete examples of some of the work that we are doing at the technological level in our laboratory. And so if I will try to have the slides. So, as we know, gene therapy has changed the therapeutic landscape in medicine. And our laboratory, which has performed pioneering research in this field with the development of gene therapy products that are in clinical trials. We are particularly interested in developing and finding new treatments for neuromuscular diseases, which account for more than 400 different diseases and altogether affect more than 1,100 people in France. So, so far, a few AAV gene therapy products have been approved, well, in fact 6, as Oumeya was saying just before, and only 2 of them are approved for neuromuscular diseases. One for spinal muscular atrophy, which was approved in 2019, Zolgen-SMA, and another one which was approved last year for Duchenne muscular dystrophy, which is Elevidys. So these gene therapy products are using AAV vectors that are derived from natural serotypes. And these serotypes have some limitations, as was mentioned just before by Dr. Adjali. So there are many challenges still for gene therapy, and in particular, for example, for the use of high vector doses in order to achieve therapeutic efficacy, and in particular in neuromuscular diseases, but as well for example, the high seroprevalence of neutralizing antibodies in the human population, which prevents some patients to be treated. As well, for example, the high manufacturing costs, which is as well a main challenge. So these limitations have created a high demand for new synthetic and improved AAV vectors that are more efficacious, that are more specific, that are safer, and that have a remaining capability to be produced at large scale and at the clinical level. So in our lab, we are trying to develop new AAV vectors with an increased potency for skeletal muscle in order to treat muscle diseases and establish proof of concept in animal models for rare diseases, in particular for muscular dystrophies, congenital myopathies, but also other myopathies. And we are using different approaches, many of them as were mentioned just before by Dr. Adjali. So we are, for that, we are using several approaches, as indicated here. We are using capsid shuffling. We are using as well computational rational design and artificial intelligence in order to generate new capsids with improved properties, in particular an increased tissue tropism, a low seroprevalence in the human, of neutralizing antibodies in the human population, but as well to increase manufacturing yields. And I'm going to give you a couple of examples of some new myotropic AAV capsids that have been developed in our lab or in collaboration with other teams. So the first ones are the AAV Myo2 and Myo3 capsids. So these capsids were developed in the context of a European consortium. And the technology that was used here is the DNA shuffling library strategy, so in vivo screening of capsid libraries in order to select the best capsids. And in addition, there was a peptide that was inserted in a specific region of the capsid in order to increase tissue specificity. So by doing this, we were able to select 2 capsids that were tested in animal models of muscle diseases, in particular X-linked myotubular myopathy and Duchenne muscular dystrophy. And we were able to show that intravenous administration of vectors composed of these capsids were much more potent. So we were able to show that by decreasing the doses by about 10 to 50 times of what is used in clinical trials, currently in clinical trials, we were reaching clinical efficacy in these animal models. And as you can see here, by looking at the molecular level, the expression levels of the mRNA derived from these expression cassettes, we found that there were 10 to 100 more mRNA derived from the transgene than when we were using a control natural vector, so an AV8. In addition, this capsid was able to as well detarget AAV vectors from liver transduction, which is particularly interesting when we are trying to treat muscle diseases in which we don't need to target this organ. So this can reduce potential side effects in this organ. As another example, I'm going to show you another capsid that has been developed in our laboratory. This was done by combining computational rational design with artificial intelligence using a bottom-up approach. So basically what was done is we were able to generate artificially new capsids, and in particular we're able to design a specific peptide that could be inserted in a specific region of a capsid that had some nice properties. In particular, this capsid was already able to detarget the liver. And by doing that, several capsids were generated and tested in vitro and in vivo. In vitro, for example, for manufacturing yields, but as well for transduction properties in specific cell types. And these capsids were able as well to improve transduction efficacy in specific models. And in particular, I can show you here, among these capsids, there was one that was the best, which is called the LICA1 capsid, that shows a high transduction efficacy in skeletal muscles and in heart. You can see here on these images of bioluminescence that is indicating the expression of luciferase activity. You can see that there is more luminescence in muscles and heart in these, here you can see, in mice that have been treated with this capsid as compared to a parental capsid that do not contain these, for example, this peptide, this specific peptide, or a natural AV9 serotype. So by administrating this vector in several animal models, we were able to show that this capsid leads to better efficacy at lower doses, so in the range of 5 to 10 to 12 VGs per kilo, we are able to see a therapeutic effect, so that's much lower than what is seen in clinical trials. And in addition, this capsid can as well detarget the liver. So it is as well a very promising capsid that was developed in our unit in the team of Dr. Richard. So altogether, I have shown you that we can use novel technologies in order to design and synthesize novel AAV capsids with improved properties, and in particular, increased potency and specificity in skeletal muscles in order to decrease vector doses and manufacturing costs. In our laboratory, we have as well a long-standing expertise in vector manufacturing, and and in the optimization of bioprocesses in order to produce a vector and improve the production of vectors at small and large scale. We have as well the capabilities to perform multi-level analysis at the preclinical level in several animal models and establish proof of concept studies with products that can be translated in clinical trials in patients. So this is to show an example of what we can do in our laboratory, which is the unit, the Inserm unit 951, which is an UMR unit located in Geneton at Évry. And we have been able to work on, as I mentioned before, to work and develop gene therapy products that are now in clinic with very promising results. So we can discuss more about that if you want later on in the B2B sessions. I will be very happy to share more information about the work that we are doing in our lab and the capabilities that are available in our lab. And I thank you very much for your attention.
Thank you very much, Anne Buj-Bello. And of course, B2B sessions, but you will also have the opportunity to ask directly your questions in a few minutes during the question and answer session after the two next short talks. Right now, I let the floor to Alexis Duvergé. He will talk about specific targeting of cancer cells by lentiviral vectors.
Good morning, everyone. First of all, I wanted to thank the organizers personally and on the behalf of my lab director who could not join us today for unpostponable reasons. Our lab, located in Strasbourg at the IBMC, is mainly focused on the biotechnological use of HIV. And today, I will focus on the specific targeting through envelope engineering, and I will present you a small portion of the long road to reach it. Classically, lentiviral vectors are mainly known to be used for stable gene transfer for ex vivo applications. It means that we can cure some monogenic disease with a specific targeting remaining on the isolation of the physical isolation of the cells. HIV, lentiviral vectors are called vectors since they are not able to self-replicate anymore. To cure monogenic disease, we can explant the cells modify them ex vivo, and re-implant them. But this is not suitable for cancer treatments. Lentiviral vector for cancer treatment must be used for systemic delivery in order to target cancer cells directly in situ. The goal is to find a way to bring toxic genes to these cells. But for this in vivo targeting, the targeting must be very, very precise with a high specificity. The target properties are conferred by the vector's envelopes. The evident solution would be to use the natural envelopes that we can find on HIV. But the problem is that they are not able to target the cancer cells naturally. So we have to modify them. But the HIV envelopes are very tough to modify without functionality loss due to the perturbation in the structure. But fortunately, they can be pseudotyped. It means that we can modify, we can replace the HIV envelope by the one of another virus. The envelope from another virus, as the alpha viruses that we use in our lab, are not more specific for the therapeutic cells that we want to target. But they are way more easier to modify than the HIV one, since they are able to tolerate the structural perturbations. So our lab is working on two disruptive approaches to target specifically cancer cells. The first one is to add a bit of complexity to the vector cells interactions by adding more ligands in the envelopes. And the second approach is to optimize the modifications by a strategy of directed evolution. The classical solution will be to graft a single motif in the envelope in order to target the cancer cells. This strategy is often not specific enough or not bring a great efficiency of the vector, but they can retain a good functionality of the vector since they are not perturbing a lot the structure of the envelopes. But despite this evident problem, we decided to choose to work with an alpha virus envelope, which is a bit off the beaten track. And we persevere in order to put two ligands in distant positions in the envelope to find a way to dock a bit better the cells in cell culture. But the question is now, is it effective, despite the structural perturbations? And we were able to target with a higher efficiency HER2 plus cells in cell culture thanks to this double functionalization process. But we obtained way more potent results on some other cancers, but I can't show you here today because they are still in the road to property, intellectual property protections. But even if we had some good result with the rational design, when we took a virus in this evolution, it will gain some mutations, mostly deleterious. It will increase the system optimization. And this is when the virus is at its peak evolution that we decide to pseudotype it without any further selection, and we modify the envelope, decreasing a bit more the optimization of the system. So the solution could be to bring a bit of evolution in this system to reach a new specificity level. Namely, we developed in our lab a system allowing to target, to evolve the viral envelope proteins directly on the vector to find a way to have a better entry and a better efficiency in cell culture. And our method is to replicate them on the target cells. So we modify the vector to make them carry the envelope sequence. They will be used to target cells, marked cells. The lentiviral vector will mutate the envelopes, generating a library. Since they are not self-replicative, we have to bring an helper plasmid to allow a new replication, generating modified vectors. And we use them again to transduce fresh cells and carry on the cycle again and again in order to complexify the library. In the end, the best envelopes should be selected and they should take a bigger part in the population. The golden envelope should be used after that for therapeutic applications. To conclude, we think that the design of envelopes should rely on combined approaches, since the rational design is not perfect. So even with good first results, who can be applied to other cancers, we should improve them by a natural process, because we are not able to think about proper modification on first try. And our lab is able to say that we can offer a comprehensive strategy to develop, modify the envelope proteins, and I will be pleased to discuss with you to tailor our solutions to your needs. Finally, I would like to thank my lab, which give me all the things I want to carry this research, and our funders.
Thank you very much, Alexis Duvergé. And the last short talk before first part of questions and answers, Maria Grazia Biferi, Spark Therapeutics, AAV vector biodistribution in large animals, highlighting the challenges associated to target the central nervous system. Thank you.
First of all, I would like to thank the organizer of the Hybrid Days for inviting Spark to provide a talk today. And thanks to the talk of the previous two speakers, I don't need to introduce gene therapy because they have already highlighted a lot of the characteristics of gene therapy based on AAV vectors. But during the talk, in this talk, I will talk about one of the major challenges that are currently existing to deliver gene therapies for neurological disorders. Because when we talk about gene transfer to the central nervous system, we know there are a lot of barriers that can be overcome by new vectors, but also by working at better route of administration to cover tissue such as the whole brain and the spinal cord. And at Spark, we are working in gene therapy research to a rare disease that is called CLN1 button disease, and I will provide some data on this program. The slides are not perfect, but it's not a problem. So CLN1 button disease is a lysosomal storage disorder, and basically it's characterized by the loss of the PPT1 gene. And when this gene is missing from the cells, we have an accumulation of lysosomal storage material in the lysosome. And this accumulation then brings to neuronal death and further neurodegeneration. It's a very, very severe disease with multiple forms ranging from pediatric and infantile cases to adulthood. And there are different symptoms such as motor and cognitive impairments, but also at the pathological levels you have also brain atrophy and visual loss. And these defects are affecting both the brain and the spinal cord. So one of the characteristics that make of this PPT1 an enzyme attractive for gene therapy is that this enzyme can, of course, is localized into the lysosome, so it can be re-expressed in a cell through an AAV genome. But then once it's produced into a cell, it can be secreted and can go to cross-correct adjacent cells, so that your gene therapy can target one cell, but then can be effective also in other cells, such as neurons and glial cells that are also affected in the disease. So this is the overall gene therapy approach that we are developing for this disease with a Spark proprietary capsid and with a genome that we have modified in order to express an engineered version of PPT1 to enhance its secretion. And we are evaluating multiple ROA in which we are delivering the viral particle into the CSF, cerebrospinal fluid. And today I will just show you the data that we are obtaining through the cerebroventricular delivery of this gene therapy in biodistribution studies in a large model. Here is the full preclinical development of this disease that you can see that starts from early discovery to biodistribution studies in mice, and in this case we use a sheep model. And then, of course, I will try to go also to few efficacy data that we have in a mouse model of the disease. So we have done this biodistribution study to understand better the properties of this vector and how it's working in a large model. And this is useful to understand how we can then translate the therapy to patients. So we have injected sheep in collaboration with the University of Edinburgh. With four sheep were injected with the AAV PPT1 vector and 2 sheep with the AAV GFP expressing vector, a control vector. And then we have analyzed the viral vector genome biodistribution, the production of the enzyme, its activity, and also the intracellular localization in the central nervous system. So these are the data showing how this vector was widely biodistributed into the brain cortex of these animals with all the different cortex area of the brain. We obtained quite high biodistribution. And what was interesting to see is that in this case, the sheep were injected in one brain ventricle, but then we could see also the expression of the vector in the contralateral side, meaning that both sides of the brain were transduced by the vector. We saw also how some other affected regions of this disease were transduced, both thalamus, hippocampus, and cerebellar cortex, and also different tracts of the spinal cord, the cortical, cervical, thoracic, and lumbar tracts. In terms of expression of the protein, we saw a 4-time higher expression of this enzyme in the brain, and it was also interesting to see how we could detect the activities of the enzyme in the cerebrospinal fluid of the brain, this giving us hopes that in future trials it's possible then to detect the expression of the protein in a biofluid as a biomarker. And here we can see also how the protein was expressed in the brain. Here, these are pictures from the cerebellum, because also the cerebellum was targeted with GFP expression in different lobes of the cerebellum, but also the expression of the PPT1 enzyme in the cerebellar cells. In terms of proof of concept, so we are using this mouse model that is lacking the expression of PPT1, and these mice they die early, they have different pathological features, and what we have done is that we have treated these mice at birth with ICV injection, and then we are following them over the time. These mice are still alive, and we are following them, but we saw here how the treated animals compared to untreated or vehicle-treated animals restore their locomotor function, and these are data obtained at 7 months of age, but we are currently following them also at longer stages, and they are still surviving, even if all the control animals are died. So in conclusion, here I showed you how we have designed a gene therapy. So we worked a little bit on the payload of the AAV vector, and this can also induce a better expression of the transgene. We have developed this combination of a capsid and a method to deliver the gene therapy to the central nervous system, and we are confirming the studies. We are working on the confirmation of the studies in terms of efficacy, and this platform will open other perspectives for gene therapies on neurological disorders that are, let's say, affecting the whole brain and spinal cord. And thank you for your attention. And we will be happy to take questions later.
Thank you very much.
Thank you.
Okay, you can have a seat. Now it's time for some questions and answers. So I will call back Oumeya Adjali, if you want to go back on stage. Anna Buj-Bello, okay. Alexis, Duvergé, and then you are. So the 4 first overview and short talks of the morning, dedicated both to AAV vectors and lentiviral vectors. So if you have questions, yes, okay. So you know how to use the mic, just push the button on. Marc?
Yes,
Okay.
thank you, Fabrice. Well, I have several questions. The first one to Alexis. I was just wondering whether you have already prioritized particular type of tumor or biomarker that would be targeted. I was just wondering, for instance, typically what would be the challenge in particular for selection of solid tumor biomarkers owing to the differential expression of this marker between healthy and tumor tissues.
Can take the mic. Is it connected? Yes, good.
The biggest problem that we have is the fact that a lot of markers are also expressed by healthy cells. But the main advantages of the cancer cells, if we can say advantages, is that they often express a lot of markers in comparison to the healthy cells. So if we manage to find a vector with a good specificity, but not a too high efficiency, we can manage to favorize the transduction of the higher expressing cells instead of the lower expressing cells. So we have to deal with the fact that our vector mustn't be too effective to avoid any off-targeting effects.
Another quick question to Maya Grazia. So how long is the expression of your transgene? And what is the duration of expression are you targeting?
Yes, so for the moment, the biodistribution data were provided after 2 months of injection, but on the studies in vivo in mice, we can still see the transgene expression at 10 months, and we are providing new data, trying to follow those animals as long as we can. But as was mentioned before, probably one of the characteristics of the AAV vector is that the vector is staying in neurons if they are not degenerating, of course, and then they can express quite longer the transgene.
Okay, thank you. Other questions? Yes.
Hi, Paul Nioy from Al-Nilam. Maria, I have 2 questions for you. Lovely data, very exciting. I'm curious about the tropism of the AAV you're using. Is it specific for neurons, or does it also transduce other cells in the CNS? And then my other question is whether you've tried administering intrathecally rather than ICV, and if that works.
Yeah, thank you. So for the tropism, we saw the expression in neurons and glial cells in astrocytes. For, let's say, talking about cells in the brain and spinal cord. The capsid then has also other targeting capacity in other organs, and this is good for this kind of disease that is a sort of also multisystemic disease. And the second question, I forgot.
Was on intrathecal versus ICV.
So we are exploring other ROA in large animal studies to see whether the ICV is comparable to other intra-CSF delivery methods. Thank you.
Thank you. Other questions? Yes?
Hi, Alain Martelly from Pfizer. It's a question for Anna Buj-Bello. You showed very nice data in terms of the new capsids targeting the skeletal muscle. Do you have any information around the ratio also between the distribution between the cardiac muscle versus the skeletal muscle, first of all? And then the second question will be more in terms of translatability, so how these viruses, new cassettes work in the higher species, actually.
Yes, thank you very much for the question. So for the first 2 capsids, AAV Mayo2 and Mayo3, experiments performed in mice showed that there is a higher ratio of vector transduction and transgene expression in skeletal muscle versus heart. So these capsids would be particularly interesting for myopathies that have some kind of pathology in the heart, or even they do not. So that's for these two capsids. And for the other one that I showed you, the LICA1 capsid, this one as well transduces more or less at the same level skeletal muscle and heart. And what is important to say as well is that these three capsids, in fact, do not, there is a very strong liver detargeting, so this is particularly interesting as well for treating muscle disorders in which there is no liver pathology, or in order to reduce potential side effects. And of course, now the question about the translation of these capsids into the clinics, for sure we need to have data in other models, and in particular, as in the field, what is currently done is studies in NHP animals, or eventually as well to use, at least to do some experiments in maybe humanized models of muscle tissue. So this can be done as well. But the best, of course, is to do some NHP studies. So this is work that should be done, or that has not been disclosed yet.
Thank you. Other questions? Yes?
Yes. Lila Ritanti for AGS Therapeutics. I have two questions. One is, did you see any neutralizing antibodies with this capsid? And the second is there then the possibility of a second injection with this capsid, or is the same as for the AAV capsids?
Okay, take the question. So for the seroprevalence, what was observed, at least for the MAIO2 and MAIO3, is that the seroprevalence was similar to the AV9. So in that sense, it is not an advantage, you know. And the second part of the question, sorry, was about?
The possibility of...
yes, re-administration, yes. So this is a big subject, a big challenge in the field, for sure, the re-administration of AAV vectors. So at this stage, we can say that, when we administrate these vectors, like we're doing a vaccine, so there will be antibodies against the capsids, and we cannot re-administrate the vectors. But of course, there is a lot of effort in trying to overcome this problem by different approaches. One would be, of course, to have capsids that immune evade these neutralizing antibodies. So this is work that is currently being done in several labs, and we are as well interested in this as well. But in our lab, we have developed as well another approach, which is to try to degrade, by using a specific enzyme, which is called IDES, to degrade antibodies against the capsids. Well, in fact, there will be antibodies against all the antibodies, but in order to have a kind of therapeutic window in which we could re-administrate the vector. So some experiments were done in animal models, and this is as well a possibility, to further develop these strategies. So I don't know if Oumeya, you want to say something about...
Want to add something? Yes?
Yeah.
Maybe?
Yeah, I think it's working. Yeah, no, I completely agree, and we are also working on some other strategies for immunomodulation or the development of capsids that could evade. Coming back to the chemically engineered vectors, what we are also looking at is whether the ligands that we are adding on the surface can also make these vectors more invisible towards the neutralizing antibodies. And in vitro data show that indeed they are less neutralized than the native vectors. So this is also one possibility. So increase the specificity and decrease maybe the neutralization. So there are many possibilities ongoing.
Yes, ongoing indeed. Beginning of a long story. Another question? Yes.
Yeah, Chantal Pichon from University of Orléans and Inserm. Thank you so much for these great talks you've done. Mia, I just want to ask you, the chemically engineered viral vector, did you have did you make a kind of economic analysis if you do this in terms of cost? How do you handle that? Thank you.
Thank you, Chantal. This is a very important question. I would say, yes, it's very important to make this economic modeling and comparison. I will not give, of course, numbers, but one advantage for this strategy is that we could use the same process strategies used for the native vectors, because the conjugation occurs at the end. Whereas when you develop a molecular new variant, in general, we need much more developments during the process, because in general we have decreased yields. And so in fact, if the process is ready, then you have just to work on the final step, the conjugation, and of course, the development of relevant analytical methods for to determine accurately the number of the ligands and what you have also as possibly free ligands.
Thank you. Yes.
Yes, so one question to Alexis. So Michel Cognier, University of Rennes on Inserm. So about your retrovolution process, what is the extent of mutations that you have, the length of the mutated domain, and the rate of mutations that is induced?
So for the mutation rate, we can adjust it by using some RT variants, for example, or by using the wild type one, which is known to be quite error prone, but not too much. And the mutation rate that we observed with the natural one is around 1 mutation at each cycle for 3,000 base pair. And we can modulate it by some modifications in the cycles. It's quite efficient, but not too much to avoid too much neutralization of the efficiency of the envelopes.
Are there other questions? No more? Yes, last question.
Hi, Gavin Badamale from Map Design. So I have a question for Oumeya. So you mentioned the various challenges of developing these AAVs. Are some of the challenges serotype specific? And also, would the chemical modification actually solve this issue? For example, you could find one which works best and then modify the tropism with chemical modification.
So I'm sorry, I didn't very. So what are, in terms of the ligand? I'm sorry.
Actually, all the challenges you mentioned, serotype specific or not?
Yes, in fact, it's related to the specificity. And also, so what we are facing with AV vector is that even if you have a natural serotype that is relatively specific for a cell or a tissue, you still have a number of viral particles elsewhere. For instance, in the liver, if you inject intravenously, which means that we need to use high doses to achieve the needed quantity in the target tissue. So it's a question of specificity, but it's also a question of detargeting the vector from unwanted organs.
And so the chemical modification, so if you can find one generic AAV that works best in terms of production or titers, etc., and then you can modify that to have.
Yeah, but the modification, in fact, is rational, is also rational design, if I can say that, which means that we choose ligands specific for receptors of interest on the cells, targeted cells, so that you play with the specificity of the serotype.
Okay, thank you very much for these questions and answers. Now it's time for coffee break. Please come back at 11. If we can go back at 11, please. And then we'll have other talks and then the round table. Thank you. Thank you very much to all of you. Thank you. We're going to start the second part of the session 1 with 2, even 3 new short talks, and after that, a new time for question and answers. And at the end of the day, at the morning, we'll have the round table, which will be another occasion for you to ask your questions. I remind you that if you want to have the whole program, detailed program, you get here a flash card, and you can take it just over there. Or on your program, maybe you can find it on internet too, if you need to get a full program. QR code. So, to start with new short talks on our topic this morning, I'm calling Anarita Miccio from Inserm, and you're going to talk about genome editing approaches for beta-hemoglobinopathy.
So good morning, everybody. And first, I would like to thank the organizer for the invitation. So basically, we'll discuss editing strategy for beta-hemoglobinopathies, which, as you might know, are diseases caused by a mutation affecting the production of the beta-globin chain of the adult hemoglobin tetramer. And again, as you might know, the beta-like globin gene are sequentially expressed during development, with the fetal gamma globin gene being silenced soon after birth, when the adult beta-globin gene is mainly expressed. So sickle cell disease is caused by a single amino acid substitution in the beta-globin chain that leads to hemoglobin polymerization and formation of sickle red blood cells that tend to obstruct all the small vases, leading to multi-organ damage. While in beta thalassemia, we have a reduced or absent production of the beta-globin chain that leads to the precipitation of the uncoupled alpha chain, which in turn causes death of red blood cell precursors by apoptosis and therefore anemia. And so for this patient, a potential solution is the ex vivo gene therapy based on the collection of hematopoietic stem progenitor cells from the patient, which are then genetically modified in vitro and then reinfused to the into the patient, where they hopefully will then give rise to normal and corrected red blood cells. And therefore, to this aim, the CRISPR-Cas9 nuclease-based strategy has been used in particular to correct the sickle beta-globin gene, as this system is able to generate a DNA double-strand break in the mutant gene that is then repaired thanks to a donor template containing the wild-type beta-globin gene. Unfortunately, however, this strategy is poorly efficient in hematopoietic stem cells, and we need to deliver a donor template, which could be complex and even toxic sometimes. Indeed, one of the 2 clinical trials using this strategy has been stopped because of the toxicity. And in addition, on top of that, we can have a toxicity due to the DNA double-strand breaks. Then another strategy to correct the beta-hemoglobinopathy phenotype is to reactivate the fetal gamma globin gene expression, which is usually silenced soon after birth. And this is because the clinical severity of both beta thalassemia and sickle cell disease patient is mitigated by the persistent synthesis of fetal gamma globin in adult life. And this happens in a few lucky patients, the one that co-inherited some mutation called hereditary persistent of fetal hemoglobin, or HPFH mutation. And therefore, to reactivate fetal hemoglobin, we and others have used the CRISPR-Cas9 nuclease system to this aim. And in particular, for example, one of our strategy was to evict from the gamma globin promoter transcriptional repressor, basically by deleting their binding site, which can be indeed cut out thanks to the CRISPR-Cas9 nuclease system. And in parallel, another strategy that is currently used in the clinical trial sponsored by Vertex and that has been recently approved by FDA and by the UK agency, another strategy is based on the knockout and inactivation of BCL11 gene, which encode for a transcriptional repressor of fetal hemoglobin. And in this case, the effect is the same, so at the end we have fetal gamma globin reactivation. And of course, this strategy is very efficient in hematopoietic stem cells, as shown by the latest clinical trial, we do not need to deliver a donor template. We're just destroying genes or regulatory regions. However, we still have toxicity, potential toxicity due to the DNA double-strand breaks. And therefore, indeed, we and others have shown that the CRISPR-Cas9 nuclease system induced DNA double-strand break that can lead to DNA damage response leading to apoptosis. And in addition, they can also cause the generation of large genomic rearrangements. And therefore, the idea in the field, if you could see the slide, is to basically develop DNA double-strand break-free strategies using base prime or epigenome editing. And in particular, I will focus on this base editing enzyme that are able to do C to T or A to G base conversion without generating DNA double-strand breaks. Trust me. And so, indeed, using the base editors, we have already demonstrated that we can do gene correction, and in particular, we demonstrated it for several severe and prevalent beta-thalassemia causing mutation, but we could also reactivate fetal gamma globin expression, in particular by generating mutation in the gamma globin promoter that destroy the binding site of transcriptional repressor, but also by generating mutation that create de novo binding site for transcriptional activators. And of course, the advantage of this strategy is that we have demonstrated that it is efficient in hematopoietic stem cells. Again, we do not need to deliver a donor template, and this time we also do not have double-strand break-induced toxicity. Just to give you an example, but you have to trust me, basically when we try to correct the beta-thalassemia causing mutation, we achieved a high frequency of gene correction with no double-strand breaks, trust me. And then we also observe a restoration of the beta-globin expression. And finally, you can check it, we also observe amelioration of the cell phenotype with a reduction in the frequency of apoptotic cells. And so we use also the base editor, as I told you before, to reactivate fetal gamma globin. And in this case, you also need to trust me that we achieve a good editing efficiency without a DNA double-strand break. But at least you can see that we could reactivate fetal gamma globin expression, especially when we create in the gamma globin promoter the KLF1 activator binding site. And we achieved the levels of fetal hemoglobin that were even higher compared to the one that we observed with the CRISPR-Cas9 nuclease-based strategy. And this led to correction of the sickling phenotype in sickle cell disease patients. And so, basically, as perspective, we would like to bring to the clinics relatively soon an ex vivo base editing approach, in particular for sickle cell disease, thanks to a collaboration between the Inserm, our Institute Imagine, the Necker Hospital, the Inserm unit RTTG, and Geneton, and also with the collaboration of the Genoter Biocluster that is fully dedicated to gene therapy. However, we are also developing other and using other editing approaches for beta-hemoglobinopathies and other hematopoietic disorder, and we are developing both ex vivo and also in vivo strategy that we think could be less costly and probably more easy to have in countries that in which we do not have a specialized unit for bone marrow transplantation. And finally, I would like to thank all the people of my lab and the funding agency and our collaborator, and especially the patient, and of course all of you for our attention. Thank you.
Thank you very much, Anarita. Now, Aurélie Bedel. Okay, you're from Bordeaux University, and you talk about how to secure CRISPR-Cas9 use.
Okay, thank you for the invitation and the opportunity to present our work. I am Aurélie Bedel. I am a pharmacist, medical biologist in Bordeaux Hospital, and I have a research activity in Moreau-Gaudry lab in Bordeaux. I have two parts for my talk. First, I would like to present a work for CRISPR-Cas9 use in oncology. You know that CRISPR-Cas9 is already used to edit CAR T-cell to boost immunotherapy in oncology, either to modify PD-1 pathway or to to make CAR-T universal, but we think that CRISPR-Cas9 can also be used to directly target the cancer cells. And for that, we don't want to do a specific CRISPR on a specific target, but either mimic a genetic disease by CRISPR to induce a cancer cell metabolic vulnerability. It is a pan-cancer approach. And in the lab, we have a long-term expertise in porphyria, which are genetic diseases. In these diseases, patients have cell necrosis in sun-exposed sites due to photosensitive metabolites. And we propose to induce this disease in cancer cells to induce a vulnerability. For that, we make a proof of principle in prostate cancer model, and we inactivate a Uros gene by CRISPR. This inactivation mimics a porphyria disease. Tumor cells are then photosensitive, and with light, you can induce cell death. And we had a patent for this innovation. You can see here the in vivo result for prostate cancer. And after gene therapy targeting Uros and only 3 illumination with light on Monday, Wednesday, and Friday, you can obtain a tumor regression. You can see here the pink tumor volume. And importantly, the mice survival is dramatically increased. So by combining gene therapy to mimic a genetic disease and light, we can obtain a spatial and duration control cell necrosis of tumor cells, which is very important to reduce side effects. We have now perspective for this work. We want to validate this approach in PDX model. We want to test it for other cancers, and we have to develop oncotropic vector to deliver specifically this gene therapy in cancer cells. And as Dr. Duvergé, we are working on oncotropic vectors to specifically target the cancer cells, and it is in good track. You can see here after systemic injection that only tumors are transduced with luciferase, and this work is in progress with the platform of vectorology of Bordeaux. The second part of my talk is about the safety, because it is important for a pharmaceutical product to be efficacy, but also safe. And in our team, we are working on the on-target genotoxicity of CRISPR-Cas9. What is on-target genotoxicity? It is when CRISPR-Cas9 nuclease induce a DNA double-strand break at the targeted locus, but the DNA repair mechanism are not those expected. And we demonstrate, we and others, that CRISPR-Cas9 nuclease can induce chromosomal rearrangement in many cell types, in many loci, and in many chromosomes. And so it is very important to understand why some cells have this chromosomal rearrangement in order to prevent it. We demonstrate recently that this on-target genotoxicity is cell division dependent. And the more the cell divide during the editing, the more damage they have. So based on this observation, we test the use of the palbociclib, which is a CDK4, CDK6 inhibitors, to block the cells quiescent in G0, G1 phase. And by blocking the cells during the editing, we drastically reduce the appearance of the chromosomal rearrangement and prevent its genotoxicity. Alternatively, as said by Anarita, if you don't want to have genotoxicity due to the double-strand break DNA, you can develop other alternative CRISPR-Cas9 system, and we are now working on the development of a new system based on a single strand break with a template import just at the good locus, at the good moment, and we obtain promising results, and we are supported by Inserm transfer to develop these tools. So for the first part, by description of the CRISPR-Cas9 genotoxicity, we decipher the mechanism, and we are now able to understand it and to have solution. It is very important to now validate palbociclib or other CDK4, CDK6 inhibitor in clinical trial to validate import system, and then and we think that it is very important to develop quality control for clinical trial for the safety of the patient. Thank you.
Thank you very much. Thank you, Aurélie, Bedel. And the last short talk before a new time for questions and answers is focused on this time startup. It's Mr. Stéphane Boissel from Sparing Vision. You're going to talk about pioneering genomics to save sight.
Good morning. And this is the very embarrassing moment where I realize that my presentation is probably a misfit, because I'm not going to speak too much about science, but more about business. How much time do I have? I was told 4 minutes. Is it more? Is it less?
Between 7 to 10.
Oh, plenty of time. Anyway, so Sparing Vision was started in 2016 as a spin-off from the Institut La Vision in Paris, and more precisely from the very fertile brain of Professor José Sahel, who is the scientific founder of the company. And the company is about doing or using genomic medicine tools in the space of ophthalmology. And we are more precisely focusing on in vivo. So we are not working on cell therapy, and genomic medicine and ophthalmology is the only thing we do. The company was started on the business principle of what we call a build-up. So we started with one asset, and through acquisition and licensing, we've built a portfolio of 6 products currently in the portfolio, gene therapy product and CRISPR-based product. The lead product, the one we started the company with, is now in clinical development, and all the pathologies that we address have very significant unmet need. We are working mostly or primarily today in what we call retinitis pigmentosa, which is the most prevalent form of inherited retinal disease, but we are going to expand in more prevalent disease, even more prevalent disease such as dry AMD or GA, for which there is, again, a very significant unmet need. In 2021, we entered into a strategic partnership with Intelia. I think in this room, everybody should know Intelia, the world leader in vivo genome editing, and probably the company that will be the first to have an in vivo CRISPR-based product approved, hopefully. And that partnership really put us on the map of the space. We have, as I said, one first product that started clinical development a year ago, and we have a second one that should start clinical development later this year or early next year. And so we expect 2 clinical readouts within the next 2 years on 2 very unique first-in-class gene therapy product. So I said I was not going to speak about science, but yet I will say a few words, because what we are doing in gene therapy is unique. Most gene therapy approaches in the eye or elsewhere, by the way, are gene-specific. In our case, we are gene agnostic. So we are developing product, and we have 3 gene therapy products in the portfolio that you can see on the slides. They are expected to work independently of what has caused the mutation to start with. And that has many advantages. The first one is that theoretically, you don't need to genotype the patient to make him eligible for treatment. I should not say that because my scientists will yell at me. So yes, of course, we are going to genotype all patients, but theoretically, we don't need to do that. And very importantly, which is less of a concern for the audience today, but it's a significant concern for the investors in general, is that when you go one gene after the other, especially in the space of ophthalmology, the perspective of a return on investment is very small because the indications are very tiny. And the best example is Luxturna, which is a beautiful product that works very well. But at the end of the day, the market for Luxturna is very small. And so it will be very difficult today to reproduce what has been done with Luxturna because we are in a context where money is more difficult to find. So with our kind of approach, we will have more or less the same pathway to develop the product, more or less the same cost of development, but we expect much more significant commercial return should the product or the biology work. And in the case of the first product, and that's the only scientific spin I will give to the pitch, is that we have identified, I mean, I did not, but José Sael has identified 20 years ago a neurotrophic factor. So he identified a gene. He split that gene into two isoforms, and those two isoforms are in an AAV gene therapy product, and that is the product that we are going to inject subretinally in patients to make sure or to try to preserve what is left of the vision at the time of intervention. So it's not typical gene therapy in a sense that first it's not gene specific, but also it's not aiming at replacing anything. It's aiming at protecting the cones through an action on their metabolism. So it's like a neurotrophic factor. It is a neurotrophic factor. So that's the first product currently in clinical development. And the second product is a product we bought from another spin-off from the Institut La Vision. It's also gene agnostic, but here we are not going to aim at preserving what is left of the vision at the time of intervention. So it's a different biology, a different transgene. We are going to aim at restoring function in cones that have lost their ability to function as part of the evolution of the disease. And the rest of the portfolio is less advanced. The first product that we developed with Intelia, the first CRISPR-based product of our portfolio, is probably, let's say, 3 years away from clinical development. And we are not disclosing the target, but it's, again, in retinitis pigmentosa. Here we are gene-specific, but we are mutation agnostic. And the idea would be in that target to use CRISPR to do single allele repression of a specific gene target in RP. That's very nice, very elegant. But if you believe gene therapy is complex, CRISPR is, let's say, leveling up the complexity to something totally unbelievable. And to make it work, we need a team of people. I'm not going to go through the resume of all these guys, but I'm only going to name two of them. Dan Chung, our Chief Medical Officer, joined us from Spark 2 years ago. And at Spark, he was instrumental in the development and launch of Luxturna, and he's now the Chief Medical Officer of the company. And every day that he's passing, I'm blessing Dan for him, being with us on this journey. And the second one is Mehdi Gasmi. Mehdi was educated at Geneton, and then he spent more than 20 years in the US. He was previously at Adverum, and Adverum, which is, by the way, going to announce today phase 2 data on their lead product, which is a gene therapy product developed in wet AMD, could be one of the first, if not the first, company to launch a product in that space. So we really have put together a team of experts in our space, independently of where they are coming from. We have an office. I mean, the company is headquartered in Paris. We have an office in Philly. We have 3 persons in California, 2 persons in Boston. And so the idea of that build-up strategy that was not only to find the assets wherever they are, as long as they fit the purpose, but also to put together the best possible team to succeed. And my last slide is about money. Of course, to do what we do, we need money. It's very costly. I like to say that from the patent to IND readiness, developing a gene therapy product or a CRISPR-based product, for that matter, takes $25 million, more or less. And then to get to proof of concept in man, you probably have to add something like 50. So 75 million per candidate. We have 6 candidates in the portfolio. Math is very simple. We need a lot of money. And we were very fortunate to raise a lot of money, not as much as what we need or we want to, but for a startup company in France, that was not too bad. We've raised 135 million euros since the beginning with the support from the shareholders listed in this slide, including BPI. And with that, I'm going to conclude my presentation. I think I went over the budget you gave me, but I'm sorry for that. Thank you very much.
Okay, thank you very much, Stéphane. I'm calling back Anarita Miccio. Okay, and Aurélie, Bedel, if you want to come back on stage, it's time for a few questions and answers, like we did this morning earlier. So, do you have any questions? One or several of them? Oui, yes, Mark.
Thanks for these nice talks. I was wondering, so clearly there is a problem of DSB toxicity with CRISPR-Cas9. So you have proposed alternative solutions. I was just wondering whether other bacterial molecular scissors have been tested. There are many others that are progressively identified. And I was wondering whether these other family of bacterial scissors showed more limited DSB tox.
Yeah. I think that's a very hot topic. So find a new Cas first because of the immunogenicity, especially because now we are moving to the in vivo. We would like to move for in vivo, and definitely there are a lot of companies and groups working on that. And definitely, I will give you just one example. We are working in collaboration with a group in Lithuania, Duvergé, which is really one of the person that discovered Cas9. He discovered another Cas, and the beauty of this Cas is that you can really make it as a base editor. And base editor, I mean, I say that they are double-strand free, but they are double-strand low. While in this case, we found a Cas that could be used also without generating any nick. So it's a completely dead Cas. And we have shown that, for example, this Cas can do base editing efficiently, but not generating large deletion. So this is it's very nice and important to study these ancient bacteria because you can discover new enzymes that are more and more potent. And of course, you have also a bit of more flexibility in terms of not patenting, but also development, really, in a company, for example.
And we'll talk about that later during the roundtable because you stay with us too. Okay, thank you. Another question? Yes.
So actually, I'm over here. Hi. Just to follow on from that question, I'm Paul Neoy from Allen Island, by the way. So Intelia have two programs that are in the clinic in humans, where the mechanism is to make double-strand breaks to disrupt the gene, so TTR, for example. So is this double-strand break issue something that's specific to certain genes, specific to certain guides or sites, or how are they able to do that safely, on the backdrop of what was discussed?
So first of all, I still use CRISPR-Cas9 nucleases, so it's definitely base editing. For me, they are better and they are safer. But I think that we are very lucky because we can really use base editing for sickle cell disease or for some beta thalassemia mutation. But just because basically we have a disease modifier. So with base editor, you can edit only one base and you can fix all the patient. But this is not the case for all the other diseases. So for all the other diseases, it's true that base editing could be safer, but still it's very difficult to develop one drug product for each mutation, for example, in the case of genetic diseases. And finally, just to answer first, maybe better to your question, I think in sickle cell disease, we have a problem of mutational burden in our cells, in the cells of the patients, which means that really, I think we have to reduce the potential genotoxicity that we cause to the cells. So we have leukemias. So we are also in a particular case, which is not so particular because at the end I discovered that there are many, many diseases in which there are high mutational burden. So I think that one should consider also the specific disease.
Thank you.
And maybe is it okay?
Yeah,
yes.
Okay.
Maybe about the DSB toxicity, it is important that for the moment we observe genotoxicity in vitro. and we don't know if there is this genotoxicity in vivo. The only description of genotoxicity and chromosomal rearrangement in vivo are when you target many loci, in particular targeting CAR T cells. If you target 3 locus with 3 guide RNA, you induce translocation in patient. There is a description in Science journal for that. But when you target only 1 locus, we don't know if in patients there is genotoxicity. For the moment, it is in vitro observation, and it is important to keep it in mind to try to prevent it and to try to avoid accident.
But is it frequent to have in vitro toxicity and not in vivo? Does it happen?
It's harder to look in vivo. It's much harder, at least in our model system. Usually, typically, we do experiment in mice in which because we look at human cells and we inject human cells in mice, then you recover few cells in mice that represent only few clones, and so you cannot really detect rare but real event. It's much harder, I think.
It is important to know that the extra large chromosomal rearrangements are in 0.1% of cells to 1% of cells, and when you edit hematopoietic stem cells, it is million and million and million of cells.
Yeah, of course.
So 1% of cells, it is a very big numbers of cells with rearrangement. But there is no demonstration that with one target with CRISPR-Cas9 nuclease, you have chromosomal rearrangement in patients for the moment. But this is the start of a story, and we have to be careful.
Right, thank you. Yes.
Yes, hello, and thanks for the nice presentations. I have the question actually as a follow-up of the discussion. What kind of in vitro cellular models are you actually using? Is it human? Is it cell lines? Is it mice? Is it
for the genotoxicity?
Yes, but in general also.
Oh, okay. In the lab, we use many cell types, cell line, obviously, but also hematopoietic stem cell from patient, fibroblasts, dermal fibroblasts. So we observe it not only in cell lines, but also in primary cell from patient.
Yeah, the point is that the cells that we use in culture are not pure stem cells, only a very small, we call them hematopoietic stem progenitor cells, but they are only a small fraction are stem cells. They are very, very difficult to isolate, to work with, so the result in vitro are quite limited. That's why what is accepted in the field, at least for now, is to inject the human cells in immunodeficient mice. And if after 6 months you still find human cells, you can say that they derive from stem cells. That's why it's a pretty, not fake, but artificial model, let's say.
Right. Other questions? Yes, Mark.
I was wondering to what extent blocking cell division inhibit DSB toxicity, and is it an approach that could be generally applied beyond the application that you mentioned?
We tested this approach in 2 primary cells, in fibroblasts and in hematopoietic stem cells. In 2 cases, when the cells are low dividing or blocked by a pharmaceutical approach like palbociclib, the chromosomal rearrangement are not present. So we think that in a quiescent cells, the cells have the time to correctly repair the double-strand break, and it is probably that double-strand break during mitosis is very deleterious.
The point is just that for some approaches, like the classical gene correction approach, you have to make the cells proliferate, otherwise nothing happens. And so, I mean, that's the problem. And even for base editor, for example, some classes of base editor, it has been shown that you have to make them proliferate. So, I mean, it's a bit a trade-off between safety and efficacy.
Okay. Other questions? No more questions? Okay? Thank you very much. Anarita, you stay with me? Okay. Thank you, Stéphane and Aurélie. And now I'm calling Citra Tauscher-Wivdeski. Okay. And Andoir, because we're going to start the round table, the final round table of the morning. And we're going to talk about, again, with you about your topics, but also other ones. And we'll go back to these two important issues that we saw this morning, AAV vectors and CRISPR therapy, then gene therapy and cell therapy. Just sit wherever. Yes, no problem. Yes. Okay, do you want to go back to me? Please closer to me? Don't be worried, don't worry. I will be great. Okay. Okay. Anna, we start with you. Andoir, you're Chief Operating Officer of Vivet Technology. And you are working on AAV-based gene therapies for rare orphan diseases like liver metabolism. Can you tell us a bit more about what you're doing to replace it in general view?
Yes. Is it? Yeah. So first, Vivet is a Paris headquarter gene therapy company. And we are developing AAV-based gene therapy for liver metabolic disorders. And we're using exclusively AAV. We have one advanced program that is an AAV to cure Wilson disease, which is a copper accumulating disorder with liver and neurological symptoms. And the candidate product is currently in phase 1/2. We have other indication, all metabolic disorders, for which we use the same AAV platform. So this is really also a company choice to have a synergistic approach, although we're not completely stuck to that. And we are therefore targeting CTX, cerebrotendinous xanthomatosis, and progressive familial cholestasis, among other indications. We're using AAV. We do additional gene therapy, I would say classical gene therapy, by replacing or adding a copy of the defective gene in the patient with functional copies. Yeah.
What phase are you exactly?
So we are in phase 1/2 for Wilson disease. The other products are in preclinical, some advanced, some still in early stages. The clinical trial is ongoing in the US and in several countries in Europe. And actually, we are expecting key data by the summer.
So you're facing the same kind of problems that we've talked about earlier for AAV vectors, like toxicity, for example?
Yeah, so we are not yet facing toxicity, but we are really controlling that tightly. And this is one of the challenges. One of the challenges with AAV, and among several, but there is the safety issue. And the safety is partly linked, I have to say that we are administering the product systemically. Therefore, there is a massive input with a huge number of particles that are infused to the patient. Something like 10 to the 15 can easily be the dose. Therefore, one of the issues that we might fear that we do not encounter, but that has been described in other gene therapy trial by infusion, is an adverse reaction due to the super high dose of the vectors. And there we are pointing to different problems. First, there is the efficacy of the particle per se. And it has been mentioned this morning by Dr. Adjali that one of the things that we need to improve is the specific efficacy per particle. The first thing, because if we do that first, that will allow us to reduce the dose. And one of the second issues that we have discovered with the time over the year is that when we infuse a solution of AAV, a suspension of AAV, actually there is true efficient particles, and there are empty particles or partly filled particles that are completely useless from a therapeutic point of view, but which are, however, participating to the elicitation of the immune response. Therefore, that means that there is another big effort that has to be put on the manufacturing and purification process in order to improve the quality. That is one big thing.
The quality, and to try to avoid too much important immune response.
Yeah. So by doing that, that will allow to be in a position of administering potentially the same dose of effective particle, but less total particle, therefore reducing the antigenic load that the body will see.
There are also some gene dysregulation, oncogenesis.
That is another, that's a thing that first might have one link with the dose. The more you put vectors in the body, the more you will have, especially when you go systemic, you will completely load the liver, unless you have detargeting strategy, as mentioned by Dr. Anna Buj-Bello this morning. But most of the time, there is a massive input in the liver. And then that means that the more you put particle, even if AAV is said a non-integrative vector, actually there is a very low level of integration that is random, and that can potentially be detrimental over the year. We don't really know that. We do not see big issue in preclinical studies in most of the animal model. But if we have to see something, that might be after 8, 10, 20 years, so sometime we don't have enough distance from the time of injection. And this is one thing that is really highly scrutinized by the regulatory agencies. And this is why actually they are prescribing more and more to have a follow-up period of minimal of 10 years. But for instance, in Germany, they are now thinking about 20 or 25 years because of this potential very long time delayed effect due to potential insertional mutagenesis.
So it means that patients will not accede to the treatment before a long time, before we're sure?
Well, they do exist. Once they have the treatment, and then once they have finished the 10 years follow-up period, then they are under the standard monitoring by their GP or by their specialist. So they are monitored for that. There might be specially scrutinized.
Maybe it could stop. If ever there are too many problems, it could stop.
What do you mean?
The treatment.
Well, the treatment is a one-time treatment. Actually, this is today the vision that we have. It's a one-time treatment. That might be another topic that we may address later on.
I think just to add something, I think it's true that we are afraid about the safety and we have to do it, but still we have to propose therapies. Now we have therapies for many diseases. Still the patient face very terrible disease, so it's not because we think that there might be something that we have not demonstrated that it could be that we don't treat patients now. I think
you have to try anyway, and we see
so
what happens.
We are, for example, for sickle cell disease, many patients have been treated successfully, and their life has completely changed. So, but of course, we also do a follow-up of 15 years. Also with the lentiviral vector, we have 3 trials of patients treated with lentiviral vector, and there is a 15-year follow-up.
And it's true that when this is the only therapeutic option, then the risk can be envisioned. And then that will be the balance between the benefit and the risk. But in a lot of gene therapy or cell therapy indication, it's not necessarily life-threatening disease. It might be diseases that are really hard diseases, but for which there are standard of cares that might be not satisfying enough. So in that case, the more there is, the more there is an existing treatment, even not satisfying, or depending on the severity and the threat to the life expectancy, the risk taken might be and will be weighed differently by the regulatory agencies. And we have to weigh the risk differently. So I completely agree with you. If this is a devastating disease with a very short life expectancy, then the balance and no solution, then the risk, the balance is in favor of the treatment. But for many other indications, we slip toward the other side. This is why we need to improve the safety profile.
So about that, we can go back to you directly, Anarita, because you talked about your pathologies, which are focused on hemoglobinopathies. And there is a treatment, which is not yours, but has been approved. It was approved, I think, a few months ago in October or
December
23, so 2 months ago, by another company, which is called Casgevy.
Yes.
That's it. I think it's, what's the name of the company? Is it Vertex?
So basically, I've contributed to the development of this treatment, but originally with CRISPR Therapeutics, that was funded by Emmanuel Charpentier with the Nobel Prize. So when I was working and did a Geneton, and then they've moved fast very, very quickly. So the first meeting was really in Paris at the Imagine Institute in 2015. So they really moved quickly, and then they collaborate with Vertex. So no, I'm very happy about that, especially because in France we have 30,000 patients that are affected by sickle cell disease.
30,000?
30,000.
Just in France, in the country? Yeah.
And so whenever I publish a paper, a research paper, or whatever, where it's written sickle cell disease, I start really getting emails from patients, and we are working at the Necker Hospital. We have 700 patients, so you really see patients. It's great if you could, they can be proposed. And I'm very happy. Now we have the early access in France, so it's not yet approved. I really hope it's going to be approved.
It's FDA approved.
FDA approved and approved by the UK agency. Here in France, we have the early.
Yet. Huh? Not in France yet.
Not in Europe.
Not in Europe yet. No.
But in France, we have the early access. So to basically, so the question now is for the doctors that we also ask is how this could then go to all the patients that are in need of this therapy, because still 30,000 patients, 15,000 only in the Ile-de-France in our region. It's very, it's a huge number of patients, and the cost
and it's very expensive.
The cost in US is $2.2 million now.
$2.2 million.
And so, I don't know. It's true that you can prioritize patients. You have more severe patients and less severe patients, but still is very challenging. We are waiting for some answer. Therefore, at the same time, I think we have to continue to do our research and continue to do our academic trial. Yes, because at least for sickle cell disease, I think it's a particular disease, but there are so many patients that still Vertex cannot cure all these patients. So the more academic groups or like companies could work on this disease.
And maybe with other strategies, because these strategies are taken by Casgevy, needs to take out cells to treat them and then to reinject, which means a lot of cells, so many cells. And sometimes it's very difficult to withdraw them and then to reinject them. Sometimes with patients, we don't have enough cells to treat. So you have another approach, which is an in vivo approach?
Yes, again, not just me, but many groups in the world that are working, especially for sickle cell disease, to do an in vivo approach, which would not require chemotherapy, because in the ex vivo approach, still we take the cells from the patient, but then we still have to make space in the bone marrow, so the patient receives chemotherapy. And for our gene therapy treated patient, most of the side effects were due to chemotherapy. Well, like that, if you do an in vivo therapy, you don't have to get rid of the stem cells, but still you have to reach them and genetically modify them. So in this case, it will be like for AAV, which I know is complex, but for me, simpler than ex vivo approach, which you really can do some injection. Sometimes we had patients at the Necker, and really, patient treated for some disease with the AAV. Sometimes they come in the morning and they are like one day at the hospital and that's it. While our patients, they stay 30 days at least. And so, this will be much easier, a much easier therapy. And of course, you can bring it everywhere in the world, also in Africa, where 6 out of 8 million patients
right now, Casgevy, you need to make several collects, for example, 5 times for one patient?
Yes, sometimes it has happened also to us that 5 times.
It's very difficult. It's difficult to live, to bear, that it's difficult for you to engineer, to work on them, and then to reinject. And I think you tried with one patient and you dropped.
Yes, for some patients we have tried we have worked a lot on the collection of the stem cells, and these are not easy patients. Still, their bone marrow is really full of bad erythroid cells, so we had to work a lot to collect the stem cells. So the collection point is critical. And for sure, also the procedure ex vivo, it has been demonstrated by many groups that if you culture the cells, they lose stemness. So, the in vivo therapy would be great, but definitely it's a big challenge, I have to say.
Okay, we'll go back to these issues. And you talked about earlier new base editors, and maybe we'll tell more about that. It's very interesting. But to finish the first part.
Yeah, I actually wanted to add to the complexity before you ask me questions. But first of all, thank you for organizing this wonderful conference. It's really wonderful to have an opportunity to talk about science and the next generation efforts. So on the topic of getting to the patient, right, with ex vivo approaches, for even a sponsor or an organization to manufacture in all geographies and get access to patients with those innovative treatments is maybe a task which is underestimated, let's put it this way. So I think the onus is on the sponsor to continue those efforts, meaning of pharmaceutical companies. And I also want to mention that maintaining a well enough safety database or a very good, robust safety database and safety reporting structure is really important for us to understand the long-term effects of those now very desired innovative treatments. But there's a huge commitment to the patient and to the community to that, because we are asked to come up with long-term follow-up studies. It was mentioned, mostly up to 15 years. Some organizations or some regulators may even require longer-term follow-up. And it's very important data we are generating here. It's very important information which helps us understand some of the issues we have been encountering with gene therapy. I've been very fortunate to be part of the development for Zolgensma, and first gene replacement therapy in spinal muscular atrophy in a very fragile patient population. And of course, we realized that we have long-term efficacy because we have one long-term follow-up study with patients we have seen now longer than 8 years.
So Zolgensma is the first successful development of a gene replacement therapy product for a rare pediatric disease, that's right? And it's been since 2019, that's it?
It was approved in the US in 2019 and in Europe in 2020. And we have now global reach in more than 46 countries.
How many patients?
We have dosed now more than 3,500 patients with Zolgensma. It's a systemic AAV gene therapy modality and one-time treatment. And with that, all the issues or all the desired features we would like to see in the next generation AAV gene therapy have been already mentioned today in the morning. And that's why it's so important to really follow up patients. We talked about potential theoretical risk of insertional mutagenesis. We have not seen that at all.
Not at all?
Not at all.
Your patients?
But
it's only 4 years, 5 years?
No, we have follow-up for up to 8 years now on patients. Okay, but it's also something which is, we have now, Roctavian and some other label had to update that into the label because it's a theoretical risk, and we have to be informing the community who treats patients with gene therapy what we know. So we have to be very transparent with that and where the science is evolving.
All right. But you also have another product after buying, I think, Avrobio. It's LV gene therapy for cystinosis, which is a rare disease, that's right?
Yes. So cystinosis, this is ex vivo lentiviral gene therapy, which we acquired from Avrobio. So we are continuing with their efforts to work in a very rare renal kidney disease patient population. It's unfortunately not a product because it's not on the market. Right. It's something which we have in development, and we are soon starting our phase 1/2 study, which means that we are looking for hopefully a way to accelerate into proving efficacy in patients soon to hopefully come forward with a approval, a conditional approval, what have it. But again, it's a rare disease. We have to be cautious. It's with all the subtleties of actually supplying this drug to patients that takes time, and we have to be very cautious how we move forward. So we have not. Avrobio has tested it in patients, in adult patients. They are all doing very well, and we have seen very great engraftment. So that is positive data, and that is really encouraging for us. But we have to continue studying that in the clinic with appropriate patients. And I think you mentioned that before as well in terms of what is the target patient population in general for such an innovative treatment. When you talk gene therapy with something which you cannot turn off yet, at least, you cannot, it's a one-time treatment, and you hopefully have a modality and approach which gets to the root cause of a genetic underpinning of a disease, then you really need to make sure that this is the right patient and the right treatment. And I, as a clinician in general, feel way more comfortable going forward with such an innovative treatment where there's really no option for patients, right? Where it's really, and then we deal sometimes with very fragile patient populations. Spinal muscular atrophy is such an example. We realize that actually the earlier we go in treating patients, the better. And so far, even very, very young patients have had actually a very good safety benefit profile out of Zolgensma. But we have to be always cautious how we go into the clinic, because those patients have not many options or no options. So we have an obligation to be very transparent with what the findings are, and whether the benefit risk to patients is really justified.
And I think on this subject, I think there is still a lot of basic research to do, at least for some diseases, for example, sickle cell disease, but also many other diseases, because we have disease modifier, and sometimes, and we know for also for hematopoietic cells, stem cell-based transplantation, that the earlier you go, the better it is. The cells are just doing much better, and the disease is not, it did not evolve so much.
Too much, yeah.
But still, you have variability between patient and patient, and so we don't know why, and still there is a huge work to do at research level, really in the lab, in basic research, to understand why one patient is more severe than the other, because we are also thinking about, I don't know, fetal gene therapy for sickle cell disease. It would be great, not having any more patients with sickle cell disease, but there are some patients that are just doing well. So how do you know? So there is a lot of genetics still to study.
To understand it, to understand better. And you're working on very important field. We said hemoglobinopathies like thalassemia, drepanocytosis, which are very important diseases all over the world, especially in some communities, as you said, with 30,000 people involved in France. And I think you also worked on this kind of indication, but finally you didn't go forward, is it right?
Which indication are you referring to?
For example, hemoglobinopathies?
I have personally not worked on this, and it's not part of our effort here in gene therapies, but we do have efforts in an oncology therapeutic area, which we have continued forward.
All right. So we wanted to stress a bit more the new base editors you're working on. Can you tell us more about that? Only one base is being changed, that's it?
Yeah, and actually,
less risky, that's it?
Actually, we are reproducing some mutation that already exist in patients, so we are really using human genetics. So from this point of view, we are pretty safe, because we are just representing the nature and the mutation that has been found in some patients who are not patients because they have this disease modifier. So that's from this point of view, it's good. And I think it has been demonstrated by many people that still base editing could be safer than CRISPR-Cas9 nuclease system. And there is BIM Therapeutics, that is the company founded by Professor David Liu, that was basically the inventor, let's say, of the base editing, that is also already started a clinical trial in November 2022. And also the group of Mitch Weiss is starting a similar trial like the one that we will hopefully start soon. And so, I mean, in general, I think it's safer, but again, we need to, it's safer, but there are other problems that could arise from base editing. So we need to still do the 2, 3, 4 years of preclinical study and process development before going to the clinics. But in the meantime, we have CRISPR.
So it's the beginning then of the strategy, but you hope that changing just one base, for example, a T in a C, could be less toxic and much easier.
I mean, it's really in terms of fetal hemoglobin expression, at least in vitro, we see a much higher. It's a bit like from when we pass from Lenti, that is also approved in clinics for sickle cell disease, to CRISPR. I mean, you can really see in the lab that, CRISPR works much better than Lenti.
So there are various issues like regulation. We talked about it. People have to be very careful, and regulatory authorities are very careful about what you're doing. They don't know what to do, and sometimes they're not very reactive. They should be very, very reactive.
Well, I wouldn't say that. I
No,
wouldn't say that, because I think...
I heard a little bit about that.
No. I think there's a lot of efforts. I mean, there's even there's constantly new guidance documents coming out. There is a consortium called Bespoke with the FDA, for instance, where we have been focusing on AAV vector modalities specifically, because the hope actually initially was that by utilizing the similar vector which we have used, for instance, for spinal muscular atrophy, we can just kind of clip and detach and clip something else on and change the transgene maybe, or the CGP, whatever, and we can move forward and into maybe an ultra-rare disease, for instance, where you have even fewer patients who also are in need of an innovative treatment. But unfortunately, as we have learned, it's not that easy. We have to really understand how we get best to target tissues. I mean, we are at Novartis specifically interested in CNS penetration and particularly reaching target tissues for some of the CNS indications. And with the current vector modality, that's just not, with the current AAVs we have at hand, we need to do better. So that's an effort which is important for us to focus on.
So delivery is really the most important problem for you.
Because it was mentioned before that we don't want to have, we don't need to utilize high doses, hopefully. We have been also working with different route of administration. So with an still systemic intrathecal approach, you may be able to reduce the vector load and have a different safety profile. That's what we're currently working on in the clinic as well with ongoing trials. But you also have to be very prescriptive in terms of any kind of monitoring strategies throughout the trials and mitigation strategies with regards to immunogenicity, the innate immune response, and hepatotoxicity, where steroids are working fairly well, but not in all patients because we have a very heterogeneous patient population.
Right. There is some hepatotoxicity, which is the first problem.
With all AAVs, we encounter hepatotoxicity, yes.
Because in general, the liver is the first target. It goes through the liver before reaching other parts of the body. That's it.
Yeah, it's a little bit more complicated.
Yeah, no,
why don't you go ahead?
If you go systemic most of the time, most of the vector will end up in the liver. That's the first thing. And then it induces elevation of liver enzyme and then associated toxicity. And this is also related to the dose. So again, the question of doing all efforts to reduce the dose. I'd like to come back to the regulatory authority position because I find them very helpful. They are very much aware. They know what's going on. They are the first who know everything because they see all the file in details. The thing that we don't know because there is secret, because this is business secret, they know everything. So most of the time when they ask questions, it's not only a position of principle. They have good reason to ask for that, and we can learn a lot from that.
But would you say that FDA or UK authorities are less prudent? Because, for example, we have an example of a product
they are
which
more
has been
pragmatic.
Approved, but not in Europe yet.
No,
so what's the difference?
They are more pragmatic, especially the UK. I mean, that's my
more
point of view.
pragmatic, but what does it mean?
That means that if we come with data and if we explain the meaning of the data, provided that they are convinced by the quality of the science, of course. But if we come with data explaining that we have explored all the possibilities and we have documented the mechanism of action, the distribution, even if we show clearly the side effect we have observed in the models, providing also that we explain why the model is representative for that and that, but not that representative of the clinical the clinical picture in human. If we have a good rationale for explaining what we have and that still the benefit should outweigh the risk when we'll go in the clinic, they are listening to that.
Why not in Europe?
Sorry?
Why not in Europe?
I wouldn't say that. We know that there are tendencies, for instance, I think that we know that Germany, they are much more careful. They are very picky and they ask more. You have to give additional proof that it will be safe, but it might be, I don't know, a cultural position.
Right.
But I wouldn't say they are blocking necessarily.
It just takes more time.
Yeah, I think I was surprised by UK. I was positively surprised by UK. I have to say, well, the difference between FDA and EMA, we already seen it with the lentiviral vector for the sickle gene therapy because we had already a therapy. We have already patient treated with lentiviral vector for sickle cell disease. And this was the therapy developed in France by academic groups and then exported to the Buj-Bello company. And the point, what's happened at the time is that now the therapy approved, the therapy of Buj-Bello based on lentiviral vector has been approved also in December 2023 in US, and we don't have it in Europe because basically at the time, 3 years ago, I think there was a discussion with Germany and France, and the price was too high probably.
But, but then the price has nothing to do with the regulatory authority.
No,
there is no relation.
Yeah.
The regulatory look at the safety and the efficacy.
No, indeed. This was fine. It's just
But the price is a
they
problem.
did not even arrive to the early access
Yeah, so
for
early
that.
access, that's another one. I mean, this is very important to have an avenue, potentially, if you have enough safety information in patients to allow access, and sponsors usually pay for that, so that's the whole story, which is wonderful, but it's also a good way to generate real-world evidence data. But that's sometimes difficult because you have to still be very prescriptive in what's the right patient. We have learned this also the hard way, right? Because if there's an effort on, every patient wants to be treated who has this disease, but may not be eligible because we have fragile patient populations. They may be suffering from an infection currently. They may be having comorbidities which don't allow that specific innovative treatment. Then we have a safety profile which is challenging. So it is either way a data set which helps us convince regulators to furthermore hone in on what is the benefit-risk profile for the individual patient and what do we extrapolate does that mean for a broader patient population in a rare disease.
But after that, let's go back to the cost issue. Because I think you said 2.2 million euros for one treatment, one injection for one of your treatment.
Ex vivo treatment.
Ex vivo treatment. Yes. Okay. So the cost is a very important issue for national countries. National authorities like, for example, Sécurité Sociale in France and other countries, of course. It's very, very difficult when you have 30,000 patients to treat. How can a country like France or other countries can address that cost issue?
Well, yes, how can the country? I think that might be a choice of a health politics. But the cost, the truth is that in practice, there is Zynteglo, I think, that has been, Bluebird Bio has just stopped,
Bluebird Bio.
even not try to go further in the discussion and commercialization, because the reimbursement, I think it was in Germany and another country, they were just far away. Mm-hmm. So they just stopped. So that means that from the point of view of the patient, that means no access.
Of course.
No access to the treatment. And it's really dramatic for the patient in need. And then there is the question of the cost. There is the question of the national politics about reimbursement, which is almost another topic, because this is a societal cost, choice and discussion and etc. But there is behind also the cost for developing and the cost of the product by itself, what we call the COGS, the cost of good when we manufacture. And I know it's less prominent for lentivirus, but for AAV and when we go systemic,
I don't think it
is just crazy. And for instance, for an AAV, and this is true numbers that I have from our situation, when we look at what would be potentially, with the current technology of manufacturing AAV, when we look at what could be the cost of a dose to go systemic for those indications for which we have had doses going up to 10 to the 13, 10 to the 14 VG per kilograms. That means that to treat less than 100 of patients, we need to have a tank of 2,000 liters, a fermenter of 2,000 liters. That is about 10 to 12 million.
Oh, okay.
So, and what can we do with that? I mean, this is what, as developer, this is what costs us the product today.
Yeah, I would say with lentiviral vector it's even 10 times more probably.
Sorry?
I would say with lentiviral vector it's much, much more expensive.
Okay,
lentiviral is more expensive?
And this is one then, because this is the other challenge. Okay. There is, and we haven't talked, we've talked about the proof of concept, the disease model, all the translational research, but there is all the technology also to manufacture the product. And behind this is also science, bioprocess and cell culture and chemistry, biochemistry.
And regulation. There's a lot of regulation, as it should be.
And we need to have also some science behind that to be able to manufacture this product at a cost that is also sustainable. I'm not saying it will resolve everything. It will solve everything in the problem of the pricing, but at least at the start, from the sponsor, if we could have a more accessible, if we could manufacture the product as a much less accessible price than the COGS, then the pricing at the end would likely be different.
I think the example that you gave before was perfect. If an AAV particle is more efficacious, you have to we can cut by 10 the cost if it's 10 times more efficient because you have to use 10 times less vectors.
Yeah, of course. So science is needed.
And I think we've been able to optimize some of those processes with the old now or approved AAVs, if I want to say that like this, but it's still not enough. I mean, it still is very, it doesn't get better. With small molecule development, you optimize, and then the cost gets really down to a minimum. So here is also an aspect of innovation, unfortunately, which just takes a lot of cost and resources and time into consideration. And because we are in rare diseases, we have the opportunity to study less patients, which means smaller studies, smaller clinical trials, which technically should mean less cost, but that not even that is true because for the sponsor, it's still very expensive. Unfortunately, it doesn't make a difference whether you study 100 patients or 200. It makes a difference if you study 200 versus 1,000 patients in terms of cost. But nevertheless, that's our some of the perspective maybe that industry can bring forward. But I think we are all in the same boat. We all want to move innovation forward. And for all, also, we really would like to accelerate access to patients safely. And all the regulations, I think it's been really very great learnings. Once you go, before you actually start your clinical trial, you need to talk, for instance, to regulators, right? So there's a whole process. It's highly regulated. We're not going out into the wild. No, just in case anybody thought about that, that's not the case. We are highly regulated, as we should be. And with your first clinical trial design, you get a lot of feedback, like, no, you can't do this, no, you can't do that, yes, you can do that, your manufacturing process is okay, or it's not, or you need to do this and this additional assay before you go into the clinic. But that's also an opportunity to learn about other efforts, right? Oh, what have other lentiviral or AAVs or other efforts done? Because based on that feedback, you learn what the current where the current field is moving. And it gets way more competitive and complex also for us. When we developed Zolgensma, we had really very small studies, and we had almost weekly interactions with the FDA. It was mind-boggling for me, coming from a drug development expertise in small molecules, where we showed up or we submitted a briefing book, but I didn't have ever this close collaborative discussion and dialogue with a regulator before on an almost weekly basis. Of course, we had breakthrough status and all of those frameworks which allow you to talk with regulators on a very constant basis, but that helps, and that collaboration is very important for all of us. I think we learn a lot about it.
Great. Okay, thank you. Do you have questions for panelists? Yes, Mark.
Yeah, I think that the point that you raised about the peculiarities of implementing clinical trial on ultra-rare disease, I think this is natural evolution of science. I mean, and we can learn a lot from what you are doing currently with this ultra-rare disease, keeping in mind that the general trend in clinical trials is to go towards more and more personalization. And now people are considering, even in oncology, that you are dealing more and more with a rare disease because of the stratification of patients based on identification of particular molecular pathways associated with a particular disease. So I think that thinking and prospectively thinking about this, I mean that it's also a way to assess the rational and added value of some approaches typically based on digital twins and synthetic arms. So I wanted to have your insight regarding this opportunity actually to propose new design of clinical trial and assessment of this molecule.
There's a lot of work going on, of course, not necessarily in rare diseases, but in general. And I think we are also now looking, of course, at more and more technology, right? There's protocol development with artificial intelligence elements, in a sense that certain pieces stay the same. How can you accelerate your protocol development? But in terms of personalized approach, I mean, there have been umbrella designs, there have been all kinds of ideas. Can you study within, for instance, a framework of different oncology subtypes, if you will, because you have this personalized approach? Can you still run a bigger study, but you have different subtypes underneath or separate cohorts and run this as one study? Would that reduce cost? Those are for sure things we can do. At the end of the day, still, it's quite an undertaking to actually start a clinical trial, unfortunately. And I think smaller companies in general do a little bit better than bigger companies, speaking from experience. And it's an evolving field, but I think we need to have also early engagement with advocacy groups, for instance, that helps always in rare disease, who tell us very clearly, you can't do this endpoint, but you can't do that. And then we have to figure out what does that mean for something what we call registrational endpoint, for instance, right? We have to have certain outcome measures in place which regulators find approvable to demonstrate efficacy as compared to safety. I'm talking very basic stuff here. It's really nothing new, unfortunately. So I would think it applies to the gene therapy field as much as to other indications.
Okay. Want to add something, Anne, about these clinical trials issues?
I think that in the evolution that I saw for the last probably 15 years in clinics is that the design are getting, even for rare disorder, getting more and more closer to what is and was expected for the non-rare pathology. I remember the first trial where there was no big question about randomization, no big question about how is the patient, is it still under his standard of care or not? The outcome were not that clear, and it was the early time. So I guess, I'm not saying it's an excuse, but there was some kind of naivety. Now, even for a rare disorder, there is a lot of expectation from the design that is very well controlled, that randomization and the control arm at some point comes quickly, maybe not in phase 1-2, but absolutely in pivotal, and it's a struggle then when it's a rare disorder, because first, by definition, there's not a lot of patients, and when you add in that, or where you discount all the non-eligibility criteria, you end up with, once you have defined your target population, you end up with a very small pool of patients. Okay. So to make a demonstration, and even not talking that much about a statistical demonstration, is just a nightmare. Yeah. But now the agencies are really asking for that.
All right, so maybe change a bit later. Another question? Yes?
Yes, Philippe Benaroche from Institut Curie. So as mentioned, the way to reduce the cost is to improve the quality of the vectors, AAV or lentiviral vectors. And this is more my field, lentiviruses. And despite much research in HIV, we are still very far from having a homogeneous population of lentiviral vectors, homogeneously infectious. So I'm a little bit worried about the potential to improve the quality of those vectors. So it was mentioned several times, so I'd like to have maybe some more hope if you can help me.
I think it was mainly mentioned for AAV, and I agree that with lentiviral vector, at least for the production of lentiviral vector, I think it can be probably improved, but still I think at least for ex vivo approach, what you can improve is the condition of the culture. I mean, nowadays, and also with the latest data, there are like people that can use very little virus because you can add molecule that can enhance transduction. So for lentiviral vector, at least for the ex vivo approach, this is something that could be envisioned, and it is currently done, for example. But for in vivo approach, there are even people are thinking about improving in vivo approach with potentially lentiviral vector, because there are some approach with lentiviral vector, but still it's more difficult, but still you can, for example, couple lentiviral vector with some peptide that improve the transduction of some particular cells, but it's easier for the ex vivo just adding a molecules, which still makes things more complex, because whenever we add a molecule, still we have to go back to the NSM or the MA to say that we changed the protocol. But at least you can really probably see very difference in the amount of virus that you use. And again, if you reduce it by 5, this means that you treat 5 patients and not only 1. While for AV, I think there is more room for improvement, probably. At least.
So lentiviral action or research is still very fundamental today.
Yeah, I think it's so, because there are like many companies also that are trying to do in vivo gene therapy with lentiviral vector, which is something that is very rarely done. They propose the liver, because everything goes to the liver, and you have permanent expression, permanent integration of the virus. You could have something, but there are other companies that are also developing, and we are also developing in vivo approach based on lentiviral vector to specifically target some cell types. It has been demonstrated that you can make CAR T cells in vivo with lentiviral vector. I think still you face the problem of off-targeting, but I think there are many groups that went back to lentiviral vector for the in vivo.
Right, thank you. Other questions? Yes.
Hi, Paul Neely from Alnylam. So I want to throw 2 other things into the mix when it comes to the cost, and I'd love to get the reflections of the panel on this. So one is the cost, not just being driven by cost of goods, but also by how much it costs you to develop the drug, how much risk you're taking. I mean, you may develop 10 things and only 1 makes it through the other end of the pipeline. And then with therapies like these, often what you're thinking about is a one-time administration of the medicine versus chronic management of that patient and the cost of that to the healthcare system. So how do you balance all of those things when you're thinking about what the medicine should cost? Because I don't think it's just a function of what it costs to make it.
Well, it's something which we think about very early in development, right? And where usually, as you said, certain efforts don't make it, or we have to terminate it because we think we have to go back to the drawing board. We have to come up with mitigation strategies. And for us, it was really not so much the COGS with our AAVs, but how can we detarget the liver so that we get access to other indications, or how can we get better biodistribution in certain tissues? And maybe it's fine with skeletal muscle and cardiac muscle for certain indications like Duchenne, for instance. But maybe you want to pursue other indications where you do think there is a developmental aspect of it, and you really need to reach CNS target tissue. So all of those considerations, and with regards to what is that going to cost, how big is the effort even in bringing a product forward, how can we get access, and how do we supply in the future? Those are aspects which we tackle very early in development, and they may break actually an effort to move forward. They may break a potential medicine to move forward, because there's only so much you can do if you focus on a specific disease area or on a specific modality, because I think all big companies have different modalities, including gene therapy, maybe a lot of small molecules or monoclonal antibody efforts. So it's a strategic decision very early on.
Other questions? Or comments?
There's
You had another question?
One question in here.
Where are you? Okay.
So that's a question for Anarita. Thank you. This is Suman Mitra here from Lille. For targeting CAR T cells in vivo and in an ex vivo expansion that normally we are using, we know the tumor environment is very suppressive, and that the traditional way of doing ACT would de-differentiating the cells in vitro, really taking them out from the system, really helps to have these memory cells and etc. But when you're targeting CAR directly in vivo, we're missing out, really targeting probably those exhausted T cells. So I'm wondering, what is your perspective about this, that really differentiating, taking the cells out from the patient versus directly targeting, CAR in the patient? What are the pros and cons?
Okay, so I'm not an expert in T-cell, first of all, so that's, I'm not sure I can answer for the differentiation
We'll
part.
talk about it this afternoon and tomorrow, by the way.
Exactly. But still, I think still there is the manufacture, I mean, still from one side I like the ex vivo therapy because it's you really can, especially for CAR T-cell, you really can control very well the quality. And still the CAR T-cells do not survive for a long time, but still it looks for me as a very safe product. But what I understood from companies, at least, is that still it's very expensive. And for the data that we have seen so far from some companies, still, at least in the limited model that we could have with CAR T-cells in immunodeficient mice, results were pretty spectacular. But again, I'm not an expert, especially in CAR T-cells.
Okay. A last question, maybe? It's okay? Great. Thank you very much for participating to this roundtable. Thank you. 12:30, great time for a lunch break. So we do go back here. Please restart at 3 p.m. Those you get time for lunch and for B2B meetings. See you later. Thank you.